So welcome to our fourth annual conference in our Tag C series, very generously funded by the National Institutes of Aging and by our local Follette School of Public Affairs, where I'm a professor of public affairs, and the local organizer of our team of chairs that you'll see throughout the day. So I'll give you just 30 seconds of introduction, and then we'll move over into our first session. So our aim is to provide a great informal environment for cutting edge research combining genetics, biology, social science. We have almost 30 presentations to see, so there's a lot of exciting new science that we'll see in the next day and a half, a mix of oral presentations, flash and poster presentations. You see some of the posters already set up, and two great keynotes that really, I think, are going to stretch further into really interesting topics of immunology and embryo selection. I want to welcome back many of you who've been here before, but also recognize a lot of great new work from new folks. And because not everyone's been here before, I just want to speak to the culture here is meant to be very welcoming. We're very interdisciplinary here, but it also is tons of expertise. If you're having trouble with your work, there's someone here in the crowd that can help and is really would love the opportunity to help push your work along. So this is a culture of direct and constructive comments, and a culture of discussion that's focused on increasing the impact of the work. I'll note that most of the content is going to be video recorded, as has been our tradition, and then we'll send it out to you all through YouTube in the coming weeks. And some final notes, just to use mics when you want it to be archived, when you want it to be recorded. And we have one mic here in between the tables for our Q&A. But I want to encourage you to meet new people, sit at new tables during the 2 days, and introduce yourself to people you don't know. I want you to provide feedback and take advantage of this great space and our community and great weather and a great new building here. So with that, I'll have Professor Liu come up and introduce the first session.
It's great to see so many familiar faces back in Madison again for another TAGC meeting. So the first session here is going to be about families. So the social and family environment we experience at various life stages is obviously very important for the social, economic, and health outcomes we have later in life. Unfortunately, geneticists are not trained to think about this. We're not trained to measure or model social and family environment in our studies. So in many human genetic investigations in the literature, we kind of just pretend they don't exist. But obviously, this is very important, especially these days. We know that genetics can correlate and contribute to a lot of social environment outcomes. And to be able to delineate the correlation and the mechanisms behind such correlation can be very important for us to better interpret the genetic associations with a lot of important diseases and health outcomes we care about later in life. And even more importantly, we now understand that the social and family environment we experience can be correlated with genotypes, not just ourselves, but also the genotypes of our family members, which can even confound the genotype-phenotype relationship we estimate in typical human genetic studies. So ever since Neil Rich convinced everybody in the field of human genetics in the 90s that linkage analysis was a waste of money when dealing with complex diseases, the entire field of human genetics has moved away from family-based designs. Instead, we started collecting independent, unrelated individuals across different families and performing association analysis based on this kind of studies. But because of the important implication of the role of family environment, in more recent years, there is a clear trend of going back to family-based design and start collecting these kind of samples and doing more careful within family analysis to better understand the mechanisms. So in this session, we have 3 wonderful talks. We're going to hear a little bit of everything I just said. Some studies want to better interpret the causal path between genetics and environment and eventual health outcomes. Some studies try to control for family environment to better understand the genotype-phenotype relationship without confounding. And so it's going to be a really exciting session. So our first speaker is going to be Silvia Barcellos, our colleague here at UW-Madison in the Department of Population Health Sciences. She is going to talk about the joint role of genetics and socioeconomic status people have at various life stages on dementia risk. So, Silvia.
Okay, perfect. Okay. Let's see if I can do this. Okay, it works. Thanks, Q, for the nice introduction. So this is joint work with Marina and Sjoerd, who are at the VU Amsterdam. Sjoerd is also here, and Leandro Carvalho, who is at USC and also here. So the kind of broad motivation for this paper is that there's a large association between, oh, thank you. Okay. Measures of socioeconomic status and health, but it's very difficult to understand what's the causality, right? Is it coming from health to SES or SES to health? The papers, today I'm going to talk specifically about the effect of health conditions on socioeconomic status of individuals and families, and the literature that has looked at this question in social sciences have mostly looked at the timing of disease diagnosis in what we call a propensity matching approach and an event study approach. So they're saying, okay, so here I'm going to talk about dementia. It's like, oh, you're being diagnosed with dementia at date X. What's your SES before? And then I kind of look at people who are like you that were not diagnosed with dementia, and I kind of compare them to over time. And I say, okay, that's going to give me the causal effect of being diagnosed with dementia. So there are different reasons why there are some challenges to this approach. One is that whether and when an individual get diagnosis is what we call endogenous. It depends on, how much access to healthcare you have, for example. And the other one is that, as I mentioned, if we believe that socioeconomic status also causes health, say poverty leads you to have worse health later in life, that's also going to complicate things with this matching and getting a control group that way. So here's just to exemplify, it shows these graphs. I look at our data, which is Dutch data, and I graph personal income and wealth by age, by whether you've ever been diagnosed or never been diagnosed with dementia. And what you can see is that even, decades before, people get diagnosed with dementia usually after age 65. But what you can see for the graphs is that even, a decade before, these people are still in worse socioeconomic status, right? So kind of disentangle this relationship is hard. And these are just kind of the same type of graph for net wealth, like the difference. So it shows you that this difference is highly statistically significant and large. So this is in percentile ranks. And we can see that people who were ever diagnosed with dementia have about 10 percentile ranks lower net wealth than those who have not. So today, I'm going to talk about dementia. Why is it interesting? Well, it's kind of on the rise due to population aging. It's very expensive disease, both emotionally and economically. And interestingly, the costs are not borne by individuals alone. Their families are also highly affected. However, as I mentioned, studying dementia is challenging. First, measuring dementia risk is difficult because you don't have a blood test that tells you do you have dementia or not, right? It's usually kind of there are large levels of undiagnosed disease. And also, as I mentioned, there's this complex relationship between ADRD and socioeconomic status. So what are we going to do? we're going to kind of investigate the relationship between genetic risk of dementia on early, mid, and late life economic outcomes of individuals, of genotyped individuals, and today their offspring. I'm going to do this in the context of the Netherlands, which have a very robust safety net system. So if you I think you can think about these problems as these estimates that I'm going to show here as kind of lower bound, right? If we think, in the Netherlands, we see effects on SES. In the place like the US, you're going to even see larger effects because you have a less robust safety net. So how do I do that? How do we do that? We merge a bunch of data sets. First, we start with the Lifelines Biobank that has genetic and survey-based data. We link that to national dementia diagnosis records, to longitudinal tax data, and to health expenditures. And we're going to have about 24,000 parents and 30,000 children. Okay. So first result, we see that the AD-PGI is highly predictive of dementia diagnosis. This is, by age, dementia, and you can see that, the blue line is the high risk, genetic risk, the yellow line is the low genetic risk. And by age 80, those with high genetic risk have about twice the rates of dementia diagnosis than the low genetic risk. But interestingly, different than the diagnosis, when you look at them at younger ages, right? This is kind of the graph that I already showed you, that people have lower incomes, people who were ever diagnosed. But when we look at genetics, we don't see that, right? It's kind of pretty much on top of each other. So what are we going to do? We're going to kind of look at these relationships over time, and I'm going to, tell you, like, we don't see much differences in the high risk, low risk, genetic risk for people when people are young. And then these differences are going to start arising at older ages, around the time when we see kind of the differences in dementia prevalence coming up. So how do I measure the genetic risk for dementia? So as some of you might know, ADRD is very different than some of the other traits we talk about, because it has a polygenic part that is somewhat, it's predictive, but it has like this big genetic part that is the ApoE, right? So we're going to kind of use both the polygenic part and the ApoE part, and we are going to then kind of, divide this into terciles, and the high risk that I'm going to show you today is going to be the top tercile. So I'm always going to be comparing top tercile according to this equation, where weights are coming from other places, with the bottom two terciles. So again, this is kind of similar to what I showed, that this genetic data can predict dementia diagnosis very well, starting from about age 65 or so, and it kind of increases over time. And if we look at survey-based measures of cognition, we also see that, those with higher genetic risk with dementia have lower cognition over time. So all good. So now I'm going to kind of look at, okay, so how does that then match into SES, right? So first, I'm going to look at early and midlife outcomes, and I'm going to compare those again in the top tercile of genetic risk with those in the bottom two terciles. And what you can see in this figure is that when you look at birth weight, father cancer diagnosis, number of siblings, years of education, cognition at younger ages, this cognition is at age 50, we see no relationship with the genetic measure, okay? We do see some statistically significant relationship with whether the father was born in the Netherlands and whether you have, your BMI, but this, we compare to the means, those are really small effects, okay? That were only significant because we have very large samples. The ones that really show up and are, highly significant is the father and mother dementia, which is, again, to be expected given that it's a heritable disease. So, okay, so then, we don't see much in kind of early and midlife. Okay, let's look at late life and look at first differences in earnings and income. And here, again, it's a similar graph. This is age, difference between high and low genetic risk. And and then we have earnings and personal income. And again, here we don't see much, right? Not much differences, not even after people kind of get older and start getting dementia. And that could be because a lot of these people are in pensions by the time, right? They're receiving pensions when by the time they're there. So if we look on household income and net wealth, which is also measures we have in our tax data, we start seeing some results, right? As people age, we kind of start seeing negative coefficients, but again, not significant. And that's true. We can kind of like do this in a regression table where we look at the top tertile for individuals age 55 to 75 versus individuals age 75 plus. And you can see kind of the effects on cognition and dementia are much higher after 75, which is to be expected, right? But we don't really see any differences in the early versus late on the SES effects. Okay, so again, not much here on SES for parents. Now let's look at offspring, right? Because I have administrative data on taxes and registries, right? I can kind of look at the offspring of the people for who I have genetic data, and I ask, as they age, how do the socioeconomic status of children vary with the genetic risk of parents? So I'm going to show you very similar graphs here. The only difference now is that this is like the parents' age. The only difference now is that the SES measure, the income and household income, is of the offspring. The age and the genetic risk is still of the parent. And what you can see here is that we start seeing again these patterns, right? As parents age, those offspring with parents that have higher genetic risk see lower income and household income. And that's even more large and significant if we look at net wealth, right? Net wealth and assets, right? So here we really see kind of this reduction of those with like high genetic risk having lower SES. Okay, and again, if we do this on a regression table, we again see these results very clearly, right? So here is for then when they are young. We don't see much, right? If anything, it's positive, some positive effects here. And when we start seeing, when they're after age 75, this is where we're seeing like the large negative effects, like it's up to, 20,000 euros smaller net wealth. And this effect is coming mostly from a reduction in real estate assets. Okay, so just going to conclude. So we have a few minutes for discussion. So we find that genetics is predictive of diagnosis on cognition on this administrative data. And while we find no evidence of significant negative effects of ADRD on genetic, on own health, SES, income, on wealth of their own person, we do see large effects on the next generation, up to 15% on the kind of reduced form increases in genetic risk. So I'm going to stop here and take any questions if there are any. So thank you.
We have a mic here, so feel free to just line up here and ask questions.
Okay, happy to be the first one. So Sylvia, so when I was, maybe it's not exactly what you are looking at, but one question in my mind is like whether people are aware of this kind of risk and do they actually have any kind of financial plannings, especially for the offsprings, right? Because if they have any knowledge, know this particular disease could be genetic, then they will adjust their expectation of how likely they will get this dementia disease, and then is there any kind of expectation effect there?
Yeah, that's a great question, and that's one that Nick Papa George and Kevin Ton have a paper on that, and they find mix, right? Like some people do, it's kind of that's my reading of their paper, and I think if anything, again, this is a context where different than the US, there is kind of a pretty general safety net in terms of long-term care. So that we are still seeing effects, even if people are planning, suggests that even they're not planning at all, they don't know, I think they know because of they see their parents developing dementia, right? But there might not be reacting to it, right? So, but yeah, it's a good question.
Thank you.
Yeah.
Hi. I was wondering if you had any thoughts about how much this might be driven the effect on offspring's economic outcomes through things like caring for parents, or spending money on care, or just the cost incurred. If there's any way to maybe try to measure that.
Yeah, we wanna get into that. We are gonna look at some auxiliary outcomes. That's kind of exactly where we want to go next. We want to look at like gender differences and look at things that we think we have some hypothesis of how, if it's caregiving, how that is. Yeah, but that's definitely one of the mechanisms that we have in mind. Yeah, thank you.
Hey, so I was surprised that the PGI is predictive of whether parents are born in the Netherlands. Again, I don't remember the coefficients are, but like when I was looking at it quickly, it was like a 0.5% or something was the.
Yeah, but you see, almost everyone is right, born in
the Netherlands. But isn't that surprising? Because if you had coded it as father not born in Netherlands, then the coefficient would be roughly comparable to the mean, right? So it seems like the PGI is like strongly predictive of whether or not you're born in the Netherlands, given that small fraction of people born outside. Like, do you know where these people are born? And maybe it's such a small group that it's won't have big downstream effects. But
yeah,
but it does seem surprising, right?
Yeah, I think it's a pretty small group. Yeah, I don't know if Stuart remembers. Right. Yes, that's one thing. But
then it's predictive even within Europe then, and that's
right. Right.
Little...
But is it a fluke, right? Like that's the thing. We are testing a lot of things and blah blah blah, right? So
I mean, you're like 2.5 or something, right?
Right.
2.25 maybe.
Yeah.
With a T.
Yeah. Yeah. But that's a good point about the inverse probability. Yeah. Yeah. Thanks. Okay.
Great. Our second speaker is going to be Tito Scalama from USC, and he's going to talk about the interaction between genes and early life socioeconomic status on a variety of outcomes people have later in life.
Thank you for the introduction. Okay.
So, so the key question is, does parental socioeconomic status, can people hear me now? Okay. Does parental socioeconomic status amplify or attenuate genetic associations? Is there complementarity or substitutability? So there's the Scar-Rowe hypothesis, influential hypothesis in social science genetics, that there is a moderating effect of low socioeconomic status on the heritability of children's IQ. And our question is, does this hold more general, also for other traits? So we're going to evaluate the interplay between genes and parental socioeconomic status as a measure of early life socioeconomic influences, the environment that an individual experiences. And the question is, are there independent effects, genetic and socioeconomic, or is there also a more complex interplay? So why would we care about this? Well, if there are environments, socioeconomic environments that have an influence on your genetic expression, then this may help with understanding if there's room for policy intervention. It may also help in the same way for early detection, in the sense that you can identify people on their or families on their behavior, and maybe you can have some policy interventions. And then you can think about fairness considerations if you have these two very large influences on life, your genetic and your socioeconomic environment. And if this is kind of random, you can think about how fair is it if people have an unfortunate draw versus a lucky draw. So the influence of genetic factors depends on environmental exposures or gene by environment interplay. There are studies that suggest this. Theory also suggests that there should be this kind of interactions, basically that disparities in health, socioeconomic disparities in health, are the result of cumulative disadvantage over as one ages. So this points at the importance of early life environment. The evidence, however, on G by E interactions is fragmented. Findings vary across traits. Most studies analyze only one outcome, and results are typically coming from a single data set. So it's unclear whether these G by SES interactions are more general or more trait-specific or more specific to specific environments. So what we're trying to do here is we're trying to provide the first sort of systematic analysis of gene by parental socioeconomic status interactions. We do this across 45 health and human capital traits. We do this in 3 longitudinal data sets that are a bit comparable. They focus on older individuals, roughly about ages 50 and above. In the Health and Retirement Study, HRS, the Wisconsin longitudinal study, WS, and the English longitudinal study of aging, ELSA. And we use a consistently constructed measure of parental socioeconomic status as the measure of experience by their children, and also polygenic indices. So why does a consistent approach matter? If G by SES is structural, it should appear across many outcomes and settings. This is an open question. And if it's more context-specific, the results may vary by the trait and by the setting. So this may help identify specific environments and specific traits with potential for intervention. So our empirical model is quite standard. We use 45 different phenotypes. That is our YI. And then we have the main genetic effect. We have a main effect of the environment, E. And then we have a bunch of controls, and we also have the interaction. So again, we use the HRS, WS, and ELSA. These are longitudinal studies. They have rich phenotypes and rich genetic information, as well as a consistently measured, comparable measure of parental socioeconomic status. They have similar age ranges, and we focus on older age. So we hypothesize that we have these big effects. We have the genetic environment, we have the genetics, we have your socioeconomic environment that your parents give you, and then we look over time at older ages when all of this genetic and socioeconomic disadvantages have accumulated, or So basically, in this way, we think we should have hypothesized basically that we have power. We start with GWASs from the GWAS catalog and the GWAS atlas. We have a bunch of criteria by which we select them, mostly for reasons of power. We focus on European ancestry, and the methods by which we select these GWASs is here's a number more additional measures. Genome-wide significant SNP counts has to be at least 6. If there's a more relevant phenotype, then we'll use that one. We drop low prevalence GWASs or traits. And yeah, if we have a more powerful GWAS, we'll use that one. We focus on Europeans, and we need the trait to be available in the 3 data sets. In this way, we came up with 45 traits. You can see them here: anthropometric traits, health traits, health behaviors, well-being, and personal traits. So then we harmonize the summary statistic, and we meta-analyze with 23andMe data if we have it, if it's available. So then we use these GWAS summary statistics that leave out the HRS, WS, and ELSA for reasons of statistical independence. We then generate 2 types of polygenic indices, the kind of standard ones, one that includes all the SNPs, and one that focuses on the genome-wide significant SNPs, the top SNPs. So our measure of socioeconomic status is basically captures sort of the socioeconomic status of individuals in their childhood. We're looking at these older people in the age, above age 50 and older, and we get an estimate of their socioeconomic status of the parents, basically. So parental socioeconomic status. To get at this, we use a structural equation model to reduce measurement error. We standardize that to be mean 0 and standard deviation 1. And the measures that we use is basically the mother's years of education, the father's years of education, and then the father's occupation in 5 categories, which are ranked by income. So then we estimate an OLS regression. We pull the 3 data sets together. We also look at them separately. And it's basically, again, the same thing. The main effect of the PGI, the main effect of the socioeconomic status variable, an interaction, and then we control for a bunch of things. So it's this linear model with an interaction. Now I'll be discussing the results for all the 45 phenotypes, and then we focus also on 9 phenotypes in particular. Okay. So here we see the first result. We see these 45 traits. If you look at the dots in red, then you see that's basically the socioeconomic status effect. At the top you have, for example, depressive symptoms, and socioeconomic status is protective of that, so you see a negative number. If you look at the bottom, very bottom, we have education. If you look again at the red dot, you see that it has a positive effect. So you see that the polygenics, that socioeconomic status has a clear, strong effects. If you look at the blue dots, those are the polygenic indices. These all have positive effects. You see that there are differences in the power of these polygenic indices. And then we see our main effect, which is that the yellow dots are all around 0. So we find no evidence of gene by environment interaction. Here we see all the data sets together, the mega-analysis and the individual data sets. Then we focus on the 9 phenotypes. I want to be quick here. Basically, we use these phenotypes at the top where we have basically a negative gene-environment correlation. So you can see that the red dots are negative. This is the socioeconomic environment effect. At the bottom, you see the ones that have a positive effect, and in the middle, we picked ones that are sort of in the middle, essentially. Anyway, the result is again the same. We see basically that there's no gene-environment effect. Here we see it in the mega-analysis. We put them all together. The yellow area is basically the interaction, which is centered around 0, while you see that the polygenic indices, they have different strengths, sort of, but you can see that they're all to the right, and the mean is roughly 0.1 for the polygenic index effect. Here we see them by data set. We see again all of these, the yellow part, the interactions around 0. Then we focus on only those polygenic indices for which we have sufficient power. So we restrict them to have greater power than 0.05, greater, coefficient effect sizes. You see no change. This is then for the top SNPs. I'm going to skip that. This is the more interesting part. And the last slide, here we see a volcano plot analysis that puts it all together. So we see in red, the red squares, if you look at the ones that go, how do I do this? Maybe I should use the pointer for this. Wait. Yes, okay. So here we see the red squares. These are your socioeconomic status variable. For negative traits like depression, etc., they have negative effect sizes, right? And here you see them, if you focus still on the red squares with positive effect sizes. Of course, what we're seeing here is that if the effect size increases, our p-values are getting better. We have a lot of power here. Now, the PGIs follow a similar thing. They're always positive. These are the triangles. And we have, in these areas especially, a lot of power. Now, if we look at the interaction effects in yellow, we see that, if the effect sizes increase, we go up again. We have the left side for the negative and the right side for positive. But basically what we're seeing is that they are, most of them, that only a few meet the 5% significance criterion, which we see here. There's 1, 2, 3, or 4 maybe. And if you do a Bonferroni correction, we find none, basically. Okay, then we do a bunch of robustness checks, and you've seen those before. This is then the discussion. So what we did is we performed a systematic analysis, many traits, 45 across 3 data sets, well-powered GWASs, precise and consistently measured families, socioeconomic status factor, and we don't seem to, we had this, notion of basically that we should have enough power by looking at early life effects much later in life. And yeah, the evidence suggests that we find no strong evidence for linear gene-environment interaction. The predictive power of the PGIs does not appear to systematically differ between individuals born into high and low socioeconomic status families. So the scar row hypothesis is not confirmed, also not in this broader sense going beyond IQ and EA, and this should be potentially of interest to the broader literature in social science genetics. I'll leave it at that.
So, I'm fine. No, because we are recording. So people are moving. No, thank you. My understanding is that in the past literature, the SE interactions are typically small and fleeting and very hard to find. Do you think you really have statistical power to detect these, given how noisy the polygenic scores are? And then could you explain briefly how you did the measurement error correction for STS? And what are your plans to correct the measurement error in the polygenic scores in the future?
Yeah, so, okay, there's a bunch of questions. So these results are potentially fleeting, and some of them in the literature that you're saying. But there is this idea still that these things exist, right? These gene-environment interactions. So that's what we're trying to study, right? So it would be consistent, right? That we're not finding anything with those sort of what you call fleeting results. But and there might be a problem in this literature actually, in the sense that there could be publication bias, right? Some of the results are positive; those can be published, and the ones where you find zero effects are not being published. What else did you say? Measurement error. Yeah, I think we need to look at measurement error. You see that in our next steps. We want to maybe tackle the downward bias in polygenic indices with genetic instrumental variable methods. That would be some of our next steps that we can do. We cannot do this for all of our traits, so we'll have to do this maybe for a smaller set of traits, because we'll need large GWASs. We need to run our own GWASs, I think. And then I think the second point is also important. We may want to explore variance GWAS. What that is basically regular GWAS is basically ask a simple question. They ask, what's the linear direct effect, right? So what we're missing is, we didn't ask in those GWASs what the variance is, and for what we're trying to study, the variance may be much more important. So we want to also explore variance GWASs.
Sorry, let me finish this first. Just very, very quick question. Maybe I missed somewhere, but how should I think about the parental investment, for example?
Sorry, what?
The parental investment, because this like SES and genes are relatively more exogenous. And in your regression, like, should I think about this parental investment or this kind of parent strategic behavior? Which part will appear or absorbed in your access?
I'm not fully understanding the question. You're saying where does the parental investment show up?
Yes.
It doesn't show up, right? It's our hypothesis that we're basically looking at the socioeconomic status captures all of that, right? It's differences between families, and we're not measuring these investments here. We're just looking at the hypothesis. Is basically, if you have more educated families, there are more parental investments. We know these kind of things, and that in the end leads to higher advantage. Does that answer your question?
We can talk later, because
there's
one more question, so I want to just keep the time.
Yeah, thank you very much for your analysis. I think these results mean a lot for people like me doing J/E analysis. And I have one probably suggestions and one question. My suggestion is that since two of the data set you are using are HRS or ELSA, so the cohort variation is pretty large. The cohort variation in the marginal distribution of parental SES is quite large. So the same level of parental SES, say high school education, means very different thing for people of different cohorts. So I suggest you consider the rank-based measure of parental SES that are recently advocated by Yu Xie, Xi Zong, Jenny Brand, or other sociologists, which may be a better measure, a more cross-cohort comparable measure of parental SES, and then may yield different findings. My question is about age. How do you think the role of age in your research? I'm not sure which wave of Wisconsin longitudinal study you have used, but HRS and ELSA are sample of older adults, among which I would be not super surprising if parental SES, which is a very far moderator for them, do not probably have weak moderation effects of the genetic effects on mental health or cognition, because for older adults, the cognition is more of a memory capability instead of a cognitive skill measure by James Heckman. So maybe age matter to some degree. And with Wisconsin longitudinal study, you may, I don't know how much measure are there. I have never used that. Probably there are some comparable measure across different ages. And maybe parental SES moderating effect is larger for younger ages than for old. So maybe age is the reason that you find few large effects, I guess. So that's my question. I wonder how you think of that. Thanks.
I think I heard 20 questions, but so let me see if I can. So you questioned the socioeconomic status variable. I want to say what we did is these socioeconomic status variables are very similar, and what we do is we do a structural equation model, right? So, and then we normalize it also. So in the sense that measure is as comparable as possible as we can make it. It's not going to be perfect, but it is kind of standardized through this method across the data sets. And then you had something about age, right? I think that's our hypothesis basically is, and I think it makes sense that this cumulative, that there is this notion of cumulative advantage. So if you are born in a family with higher education and more money, higher socioeconomic status, there is this idea that these effects accumulate over time, and the early life effects are known to be strongest, but your parents are still with you often much later in your life, and you don't need your parents anymore. You're in this other. You're also more likely to be yourself of higher socioeconomic status, right? So this in our mind was a proxy of the biggest kind of social environment that you have, basically, which is that of your parents. Yeah. It was him answering long.
We can follow up later. Yeah, it is 20 questions. So we can follow up later. Let's thank our speaker again. It's time to move on to the third talk in the session. Our speaker will be Dinoza Muslimova from VU Amsterdam, and she's going to talk about how controlling for parental genetic confounding can lead to better estimate for the causal effect of personality traits on various socioeconomic outcomes.
Hi, everyone. Thank you for having me here. So this is the first time I'm presenting this paper, so bear with me. And it is in collaboration with a few amazing co-authors. Many of them are here today. So I'm going to talk about, this is a slightly revised title. I'm going to talk about the economic consequences of personality traits, evidence from genetic variation. So what is the broad motivation for this paper is that socioeconomic outcomes exhibit substantial heterogeneity across individuals. And this heterogeneity is very important for designing effective policies and just understanding this socioeconomic traits in general. And personality traits, and this is coming from all the literature on different contributions of cognitive and non-cognitive skills, and personality traits, which are part of that relatively stable patterns of thinking, feeling, and behaving, can be partially explaining this heterogeneity in socioeconomic outcomes as well. So we will mainly focus here on big 5 personality traits, which are openness, conscientiousness, extroversion, agreeableness, neuroticism. And those are usually widely used in various human resource management practices and are replicable in different contexts. That's why we focus on this particular personality traits here. Normally, estimating their causal effect is hard because we've talked a lot already in the first 2 presentations about that today. They're usually correlated with parental background, different socioeconomic characteristics of parents, etc. So that's the challenge that we try to address in this paper. And on the other hand, at the same time, while this is very endogenous, I'm going to use economics terms sometimes because I'm an economist by background. So because this is endogenous in the background, direct experimental manipulation would be interesting to see to disentangle this causal effect of personality traits on outcomes. However, this is not very easy because it's very hard to come up with an experiment where you manipulate somebody's personality and then look at the effects of that personality on their outcomes. So this is a fundamental identification challenge in this literature. So what we do in this paper, we leverage random within family genetic variation that we can take out when we control for parental genetics. And we will work a lot with the setup where we look at the phenotype of this reference individual while we look at factors such as the genetics of big five of that reference, plus also the genetics of big five of reference's parents. And then we're going to use additional genetic variation for various other mechanisms that we think are appropriate to control for the channels that lie in the effect between the personality PGIs and the socioeconomic outcomes. So we construct these PGIs for the Dutch Lifelines Biobank using the most recent Schwab et al. Personality traits GWAS weights. The GWAS samples there vary from 600,000 individuals and more up to 1 million, I think. And then the SNP heritability there is also quite meaningful. It varies between 4.8 to 9%. And then the R-square of polygenic scores is from 1 to 3.6%. We impute the parental PGIs using the SNIPER method from Young et al. 2022. Then we merge all this data to Dutch administrative tax data from Statistics Netherlands and tax authorities. And then we use the PGIs for relevant traits to understand the mechanisms and confounders. So let me get back a bit into identification challenge here is just to reiterate that effects of big five personality traits may be driven not only by personalities themselves, but because the underlying genes are pleiotropic, they affect other channels as well. So we need to think about it more carefully. While we are still interested in the effect of personality traits themselves, so we're not interested purely in the effect of PGI, we are interested in the effect of personality traits on the SES outcomes. So what we do here, we use this random genetic variation that's coming from conditioning on parental PGIs. We use that random genetic variation to disentangle different channels. So first, we use that variation. Second, we in quasi-experimental settings, we know that testing for those additional mechanisms that contribute to formation of socioeconomic skills apart from personality traits, they require valid instrument for each channel. So in here, we use our understanding of how genetic variation works. We rely on this Mendelian inheritance, which provides multiple sources of exogenous variation. We know that the underlying SNPs are the same under the polygenic scores, but the weights and the variation that's remaining after conditioning on parental respective PGIs, they will provide us with this exogenous variation. And just to reiterate that, PGIs combine the sources with different weights. So the variation will be coming with the differences in the weights. Let me see. Yeah, I talked about that already. So just to give you some idea of what these personality traits look like and what defines them, we look at openness, that's receptiveness to new ideas, approaches, and experiences. Then there is conscientiousness. It's more about capacity to organize, complete tasks, and work towards goals. Extroversion here is preferences for social interaction and lively activity. Agreeableness is more selfless concern for others, trusting and being cooperative. And neuroticism is proneness to experience unpleasant and disturbing emotions. The data is Lifeline's Dutch Biobank. I will quickly go through this, not spending too much time. What we do, there is 78,000 people who are genotyped, and out of them, 35,000 have siblings or pairs of parents and children for whom we can impute both of their parents' PGIs. Then we also have survey-based measures of conscientiousness, extroversion, and neuroticism. You can see some example questions over there. And then there is also self-reported years of education and cognition in the data set to test more additional channels. And we have income and wealth in percentile ranks. That's what I'm going to show to you today, but I'm also very open to suggestions on what else do you think is interesting to test, because administrative data is very rich. And we refer to the focal lifelines participant as the reference. I already mentioned that. Just to show you some of the results that we have in the paper is first we check whether conditioning on parental PGIs helps us to isolate that random variation. And we check that by looking at the effect of the PGIs of the reference. So, these are the PGIs of the reference only. I'm not showing any parental PGIs here. And what we do, we look at the two specifications here. So we test how the wealth of the parents is correlated with the PGI of the reference for respective PGIs for respective personality traits. And then we see that when we only look at the without any parental PGI control, we see that for some personality traits, there is some correlation with parental wealth. But once you control for PGIs of parents, that correlation disappears in two ways. Basically, it becomes statistically insignificant, but also it is meaningfully around zero. So, which just confirms that conditioning on parental PGIs helps to isolate that exogenous variation. Next, we test in our data how reliable are personality trait PGIs. And we do it by regressing all of the personality trait PGIs on each of the survey-based personality measure. And what we see here is that for the corresponding PGI to the personality measure, it actually really shows up, even when you're controlling for the PGIs of other personality measures. For example, for conscientiousness, while controlling for parental PGIs, so this is already causal in the sense, the predictive one standard deviation in the PGI for conscientiousness increases conscientiousness by 0.10 standard deviations, and the results are similar for other survey-based measures that we have. Yes, thank you. So basically what we have here is that for PGI for extraversion also pops up among other PGIs for extraversion itself, and the coefficient is also meaningful, and for neuroticism as well. Right, so these are the main results, and then we start going deeper into why do we find these results. This is to address our main question, do Big Five PGIs causally affect, Big Five personality trait PGIs causally affect income and wealth? I have two main results here. This is number one for income, and that's number two for wealth. And what we do here, we regress Big Five personality trait PGIs on individual income percentile ranks in that year and for that cohort, conditioning on parental PGIs. And what we see here is that among all of the traits, neuroticism pops up quite a lot. So it basically drives 1.3 percentile ranks down in income distribution, one standard deviation in increase in neuroticism. And for wealth, we see two interesting results emerging here. One is for conscientiousness, and another one for neuroticism. All right, so just bear these results in mind, and let's go to the next set of results. Further in the paper, we do multiple exercises to try to isolate different channels through which these effects of neuroticism and conscientiousness are going. And we look at different available potential mechanisms that we have in the data set. One is education, another one is I don't have the results for other things here, but I will try to quickly go through these exercises over here. So what we see is that, for example, when we look at the regression of neuroticism PGI on neuroticism, survey-based measure of neuroticism, when you were controlling for education PGI in different ways, using education PGI itself, or using GWAS-wise subtraction, where we subtract EA from the GWAS itself, the relationship between neuroticism PGI and neuroticism doesn't change much, which just shows again, confirms again that neuroticism PGI is pretty capturing very well neuroticism itself here. And then what we do in the next set of results is that we look at the previous relationship that I showed you here, and then we control for EA-PGI. We think cognition might be also one of the potential mechanisms over there, and then maybe major depressive disorder as well. So we control for PGIs of these traits, and then we see how whether this drives any changes in our results. And over here, you can see that neuroticism is pretty stable across various specifications, although we try to shut down these alternative mechanisms. And then we do the similar exercises for conscientiousness as well. And then you can see that although maybe the power the power for conscientiousness might be affected a little bit, but the effect sizes are also pretty stable for net wealth. And then there is something going on between neuroticism and major depressive disorder, but the question still remains whether depressive disorder is consequence of too high neuroticism or whether it's a separate trait by itself. So just to wrap up quickly over here, what we do in this paper, we'll use random genetic variation in what we find over here is that random genetic variation in neuroticism negatively affects income and wealth. And this relationship persists even through various sensitivity checks. And effects on wealth are somewhat sensitive to depression. And as I mentioned already, depression, we are trying to understand what depression is exactly doing here. And then random genetic variation in conscientiousness positively affects wealth, and the effect sizes are pretty stable across various sensitivity checks over here. And what we try to show in this paper is that for controlling for alternative PGIs removes components of variation that affects our outcome through various pathways, while retaining sufficient variation specific to personality traits to estimate its effect here. What we plan to do next is we are thinking to look at other mechanisms, as long as it's possible in the data, or another data set, maybe using some other Mendelian randomization methods, but it's not the main interest of the paper to go too much into methods. I think the results are intuitively quite consistent already. And then we are thinking to look at other outcomes as well. What is in the path between this socioeconomic effects and the personality traits? It can be maybe there are some occupational choices people make, or other maybe people perform better in specific school tests, etc. So these are things that we can still add to the paper. And then the last one, we want to check heterogeneity in these effects in themselves. Do effects vary with gender or socioeconomic background, etc. So that's it. If you have any questions, let me know. Thank you.
A very small clarification question. So the outcomes were income and wealth measured in percentile ranks. The personality PGIs, what scale was that? Was it standardized or some other units?
We standardized everything.
Thank you.
Yeah.
Any other questions? So I was just thinking about how. So you've done this design with PGIs. You can, in theory, like ask similar questions by looking at like genetic correlations between these outcomes. And I was wondering how your estimates, how they compare if you would just looked at the literature for genetic correlation estimates, and maybe you could even infer like partial genetic correlation estimates, which would be equivalent to like what if you're controlling for other things. Have you looked at that? Does that exist?
Point. I think. We need to look more at the Schwaber paper for that and the previous personality trait GWASs to see if there is any kind of gene environment correlations already measured. But or otherwise, I should look at it myself. Just try to estimate it in the data. Yeah. Good point.
Hi again.
Hi.
Thanks for the presentation, Dilnoza. Is this project more about the big 5 personality traits or about the PGIs?
That's a good question. I said in the beginning that it's not our intention to focus on the results on the PGIs, but we're using PGIs heavily to understand the relationship between the two. How I think about it is PGIs provide you with this exogenous variations. Think of any other experiment that would provide you with exogenous variation in personality and how that exogenous variation in personality traits then relates to the income and wealth. So yes, I talk a lot about PGIs because we need to understand these methods more and the channels and how we work around them. But the question that we are addressing is how personality traits affect income and wealth. Yeah, thanks. That's a really good point.
Thank you very much for this great session.
All right, everybody. Well, I think we're going to go ahead and get started with the next session. Okay, well, it's nice to see so many familiar faces in the room. If we haven't met, my name is Lauren Schmitz. I'm an associate professor here at UW-Madison and part of the Social Genomics Initiative and group, which has been really exciting. And yeah, this session is all about DNA methylation and other advances. It's a flash session, so each presenter is only going to have 5 minutes to give you their big picture overview. And then after each of them gives their 5-minute presentation, we're going to come and look at their posters. So hold your questions until the end, until we can see their posters and talk to them. And we're going to go ahead and get started first with Arthi Bhatt from the University of Minnesota. Arthi. All right. Thank you.
Alrighty, hope everybody can hear me online. It's great to be back at the Advances in Social Genomics conference, and I'm excited to talk to you about some recent work that I've been doing at the University of Minnesota. So this project is entitled Housing Insecurity Across Childhood and Adolescence and DNA Methylation-Based Biological Aging with Evidence from the Future Families and Child Wellbeing Study. And this is conducted in collaboration with Nick Gratz, Bharat Thiagarajan, Mike Esposito, and Teresa Osipuk at University of Minnesota. So to set the stage with housing insecurity among youth, currently about 1 in 6 U.S. children experiences some form of housing insecurity during their childhood. And 5% of households with, I'm gonna move over here, about 5% of households with youth aged 13 to 17 report homelessness experiences on an annual basis. Additionally, about 2 to 3% of high school students report unstable housing in any given year. And patterns of housing insecurity that begin in infancy have been shown to be associated with continued instability and poor health outcomes in adolescence. And we do know with current literature that housing insecurity is a common life course exposure with important implications for health and biological aging. And we know that one of these forms of biological aging is epigenetic age acceleration, where exposure to stress can influence the epigenome, which regulates when and how genes are turned on or off in response to environmental conditions. One of the main factors of epigenetic aging is DNA methylation, which is a modification in which a methyl group or a carbon atom bonded to 3 hydrogen atoms is added to a cytosine-guanine dinucleotide sequence. And these patterns of DNA methylation across CPG sites form the basis of epigenetic clocks, which estimate one's biological age, leveraging methylation signatures associated with the aging processes. And so, oops, I think I slide was skipped here. Okay, well, I used the Future Families and Child Wellbeing Study. So I took data from ages 1, 3, 5, 9, and 15, in which participants reported whether they had experienced housing insecurity, including missing a rent or mortgage payment, experiencing homelessness, being threatened with eviction or foreclosure, or experiencing eviction or foreclosure. And we we calculated a cumulative measure of housing insecurity across waves, in which a score of 0 would indicate that they didn't experience any housing insecurity between ages 1, 3, 5, 9, and 15, and a score of 5 would indicate that they experienced at least one form of housing insecurity across all 5 waves. And including covariates of household income and race and ethnicity, we conducted a linear regression analysis using DNA methylation-based clocks at age 15. And so preliminary results indicated that with each additional wave in which a participant experienced his housing insecurity, there was about 0.24 years or 3 months of additional epigenetic age acceleration as accorded to the Horvath clock. And further analyses and interpretation need to be conducted in order to better understand why we didn't see significant similar associations in other clocks. And further analyses will also explore alternative measures, methods of measuring cumulative housing insecurity, including utilizing latent class models to examine trajectories of housing insecurity, such as participants who never experience housing insecurity compared to those who experience earlier in childhood but not later in adolescence. Housing insecurity experiences, those who have chronically high levels of housing insecurity, and those who experience more housing insecurity in the later adolescence period compared to the childhood period, as well as potentially looking at item response theory in order to differentially weight housing insecurity items in a scale, and using a high-risk threshold approach that differentiates participants who experienced 0 to 1 housing insecurity exposures across waves compared to those who reported 2 or more housing insecurity exposures. And here are some references, and please feel free to contact me at this email address. Thank you so much.
Yes, great. Hi, everyone. I'm Guillaume from McGill University. So yeah, this is our project today. So we're going to talk about something that a bit more clinical than what was done before, but a nice change of beat. And I'll be right there if you want to speak to me at poster session. So we're going to talk about dermatophyte infections, not the sexiest topic to talk about, both literally, but also it's a bit it's not discussed very much in the genetics literature, but it is the most common fungal infection globally, about 20-25% prevalence overall, so extremely common. For those who don't know what dermatophytes are, basically they're fungi that feed off of keratin on your skin, on your nails, on your hairs, of course. So extremely prevalent. And of course, as you can imagine, because we always like to highlight this, but it's a real problem, antifungal resistance is rising. And for most people, it doesn't cause severe infection, but the very last slide, I'll speak about a patient of mine who had a very debilitating disease, and that was the motivation for doing this, basically, because we hypothesized that the host genetic architecture could play a significant role in how we could maybe help treating this disease. And so our approach was the usual, really in genetic epidemiology. So Genomics association study. So we tested 10 million genetic variant throughout the genome, meta-analyzed that across about 240,000 cases and close to 2 million controls in the 5 usual kind of continental ancestries, whatever that means, just anyway. Continental ancestries. And then we use the usual approach of causal inference, so Mendelian randomization. For those who are less aware what that is, it's basically statistical and instrumental variable analysis using genetic variant as the instruments. And then we looked into whether or not we could actually use this to guide us in treating this patient. For those who are interested, really quickly, we can't see it too well, but over there, you see the Manhattan plots. We found a bunch of loci, but we're not going to concentrate on that just today. We're not going to go through all those genes. The point is we found quite a few genes whose expression or protein level in the circulation was actually associated with worse or poor risk, worse or better risk of developing dermatophytosis. Just want to highlight 3 basic pathways that we found, the last one being the most important for our patient. First one, skin barrier, not exactly surprising. It's a disease of the skin and the nails and the hair. So we were happy to find some genes that are clearly part of the skin barrier. So filaggrin, FLG, LC cluster. The KRT is actually keratin 1, but I can tell you there's like more than 40 keratin genes, and most of them were actually associated with dermatophytosis, which makes sense, right? They feed off of keratin, so that makes sense. Immune regulation. The only thing I'm going to say here is those are not the usual immune system genes that we find in other like disease like COVID or pneumonia and all that. So we were a bit surprising, but they're not, like many of them are actually known to be involved in immune processes on the skin. So that was very nice. The last one is actually retinoic acids. People who don't know about it, it's vitamin A. So we found a few genes that were very interesting here. RARA actually regulates RA signaling, but FTO, which is a gene very well known for obesity, and obesity is a big risk factor for dermatophytosis, we thought we could basically make a link between FTO, obesity, and dermatophytosis through RARAS2, which is important in the signaling of retinoic acid. So that brings us to the last slide, which was basically the patient to me in my clinic that motivated most of this work. This was a patient, I shouldn't say compound heterozygous. He probably has more of an haploinsufficient CARD9 deficiency. That's probably more what he has. And he has been stuck with basically whole body trichophyton dermatophyte infections. And so basically, he had run out all of the options in terms of antifungal. He had become, like, the fungus on his skin had become resistant to all of this. We tried a topical tretinoin, no real improvement. But then we tried also oral isotretinoin, which is also known as Accutane, used for acne. Within 1 week, he said his main symptoms was pruritus, so itchiness, whole body basically disappeared. Here, those are the picture. I feel bad because this was during the winter and this was during the summer. So of course you kind of amplify with the tan the difference. So this is a bit of a biased picture. But you can sort of see that a lot of this is actually better than what it used to be. But it's true that this is biased because of the tan. But yes, so basically come talk to me afterwards.
Yeah, thank you so much for coming. I'm Yusin. I'm a fourth year PhD student at Princeton, and this is part of my dissertation project. And yeah, any questions or comments are really welcome. So usually when I present this project elsewhere, the question that I get asked is, why genetics? But I guess today here, I'm probably expecting more of a question of why migration, right? So let me try to briefly situate you for why I'm interested in migration and why I'm using genetic data to do that. So the overarching motivation is just the fact that migrants are just super heterogeneous group, right? It's not a unified group. They have very heterogeneous outcomes, and I just wonder why. Like, is it because they're like differential selectivity into migration, or is it some sort of like differential treatment on migrants, right? And there are two models for migration in general. The posit model argues that migration really requires a lot of determination, a lot of resources like time, money, all kinds of things. And therefore, they say that it leads to better outcomes and are positively selected. While the negative model argues that migration really disrupts your daily routines, your social ties, you're in a new environment, you have to adjust, blah, 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 and therefore leads to worse outcomes and therefore usually are negatively selected. And I argue that those two models are not really contradictory because they really are operating based on very different sets of assumptions. So the posit model are more thinking about the long distance voluntary migration, those who are like by their own will selecting into migration, right? While the negative model are more thinking of the compelled movers, like more of those who are evicted, who are, you know, housing insecurity, right? And therefore they're really looking at different groups of migrants. That's my argument. But unfortunately, a lot of cases, the researchers are unable to identify between those two, partially because of data limitation. They just don't have enough data to really, especially pre-migration traits, to really differentiate, oh, what's the difference between those two groups of migrants and which sets of assumptions should I adopt? They just arbitrarily choose one of those models and just go from there, right? So I argue this is where genetics could go into the picture, where some of the previous studies using genetics to study migration, a lot of authors are sitting here, right, argue that genetic profiles can really serve as additional data points where, because they don't change, and therefore even if they're collected after migration, they can still provide some insights about pre-migration traits, which is exactly what we need for selectivity. So those are my main results. Basically, I'm using @health data, especially wave 1 to 6, and I'm separating between movement between wave 1 3 and movement between 4 and 6 because the first waves are more before the college age-ish, and the later ones are like later life out movement. And I argue that the left is more like parent-driven, while the right is more self-driven migration. And again, I'm looking at the distance. Basically, is there like a systematic difference between the genetic profile of short distance migrants and long distance migrants, where I'm defining short distance migrants as those migrated lower than 5 kilometers, which is about 3 miles, which according to the residential mobility literature, this is the usual distance within a block or within a neighborhood where like people who are evicted or forced to migrate usually move. While the long distance migrants are over 100 miles, basically, which is, again, according to the American Community Study, 80% of migrants are within this distance. And the results, basically, the overarching idea is that, yeah, there is, like, in general, more positive selectivity for health, behavior, and I think, yeah, educational attainment outcomes. We can see that one is most significant, while there is a negative selectivity for mental health in general. And there are some nuances and differences I'm happy to talk more about if you come to my poster over there, the orange one, Princeton color. Yeah, so I also have like other findings, again, just like testing the heterogeneity among different identities, like intention to move, neighborhood characteristics, parent, individual SES, and gender. Happy to talk more. But the main takeaway here is just that there are a lot of cases more heterogeneity among migrants than between migrants and non-migrants, and therefore I argue that we should really care about what kind of migrants are you talking about instead of like just saying did you move or did you not move, right? Some of the future steps, which is going to be the other parts of my dissertation, are going to look at more the outcome part, just see whether the traditional migration outcome research really missed some sort of selectivity based on genetic selectivity that I found, or and also using some of the within family models to really try to identify some of the causal effects of such selection. Yeah, thank you so much. Yeah, happy to talk more.
Hi, my name is Elijah Watson. I'm a PhD candidate in anthropology, also doing an MPH in epidemiology at Northwestern University. Today I'm going to be talking about the Cebu Longitudinal Health and Nutrition Survey. This is a long-standing birth cohort study that's started in the Philippines in 1983, and it's followed the mothers and their offspring through to today. We're about to do another survey wave later this year. There's been a lot of great work done from this study on growth and development, nutritional epidemiology, development economics, but something that's yet to be examined is the kind of like political economic context of the year that they were born into. What was happening in 1983 in the Philippines? So to give some broad background, a decade earlier, this is when Ferdinand Marcos, the president of the time, declared martial law in the Philippines. A key figure, Ninoy Aquino, was also imprisoned that year. He was in prison for 8 years. And 8 years later, he was allowed to return to the U.S. or leave from the Philippines to go to the U.S. because he needed heart surgery. So he was given political exile. But he decided in May 1983, he decided in August 1983, this Cebu study I'm talking about began enrolling in May and went to the next April of 84. But in August of 83, Aquino decided to return to the Philippines. Marcos, the dictator at the time's health was failing, and he decided this might be the time to return, even though he was advised not to return. He knew his life was under threat. Ultimately, when he stepped off the plane on August 21st, he was assassinated, and it sparked really rapid political and economic effects. We can see inflation rising really rapidly after the assassination. There was international news coverage, and in Cebu, which is in the central of the Philippines, in the Visayas region, we can see metropolitan Cebu here. There were 33 neighborhoods that were randomly sampled, and they collected data on all the pregnant women in those neighborhoods. We can see the black are those that are outside of central Cebu City, and the ones in purple in the middle are in central Cebu City. And the reason I draw out that comparison is because central Cebu was really differentially affected by the crisis, both economically. We can see a big drop in household income over 83 to 86 if they were in Cebu city center versus elsewhere. And also it was the center of political mobilization. There were a lot of protests around the capital, particularly in this area. So the research question I ask is, did in utero exposure to this post-assassination political economic crisis affect epigenetic aging 4 decades later? And we're looking specifically at the interaction to see if effects are dependent on residing in Cebu city center versus outside. And then we break it into late, middle, and early gestation. This here starts a little blurry. This is looking across different epigenetic clocks, but what I want to highlight now is that we see increased epigenetic age across all of them, particularly for middle gestation exposure if they were exposed in Cebu City. I have a lot more to talk about at the poster, particularly there's in the background an El Nino drought going on, but we show some evidence that seems to suggest that it might be leading to our estimate here being conservative. And also, these effect sizes are quite similar to work from Lawrence Schmitz and others looking at the Great Depression, fetal exposure, and also the Dutch hunger winter, even though we're looking at quite different shocks across these populations. Thanks.
Okay. Hi everyone, I'm Luin Zhang. I'm a 4th-year PhD candidate at Princeton, and I'll be on job market this fall. So this is joint work with my advisor Sam Trejo, where we asked how we could measure skin tone more consistently and reliably in social science data, and we aim to provide complementary tool that expands existing measurement strategies, not a replacement for current measurement. But to take a step back, why we care about skin tone? So a lot of the literature on racial inequality have tend to focus on disparities between discrete racial categories, such as black-white academic achievement gap. But these could obscure within race inequalities, and researchers are increasingly interested in measuring stratification along continuous visualized characteristics. So these are like individual level continuous traits that can vary both between and across and within racial categories. And this matters because individuals with the same racial identity can still experience race very differently. And among these kind of visualized traits, skin tone is actually one of the most well-documented one. So prior research have shown that lighter skin tones are always socially advantaged over darker skin tones. And skin tone has been linked to a wide range of valued outcomes, such as education, health, and perceived attractiveness. But there is a very important measurement problem. In most social science data sets and biomedical data sets, skin tone is not collected at all. And sometimes even when it's collected, it's usually like a single rating reported by a single interviewer at single time point. And if these single ratings are noisy, then maybe the field has been underestimating the true magnitude of skin tone stratification. So that's why we are interested in this and want to do this kind of study. And our study has three parts. So first, we filled a novel online survey where we asked the respondents to rate the skin tone of this standardized photo like this using this 5 category scale that's the same as the ad health. And then this allows us to quantify inter-rater disagreement and to know to what extent these single ratings are noisy. And then also the core of this study, we build a genomic prediction model among those non-Hispanic Black and Hispanic participants in ad health. So basically the approach uses local ancestry information around those pigmentation related loci and also machine learning strategy. And it's in spirit very similar to the idea of admixture mapping. And third, we provide an example of substantive analysis using these predicted skin tone measures to see how it's useful in data sets that even without direct measure of skin tone. And here are the results at a glance. So basically, we first noticed that there were substantial disagreement in skin tone ratings that's also reported in existing literature. So for example, the subject level ICCs only are only about 0.34 to 0.42. So among these Black and Hispanic subjects being rated. So basically it means a large share of rating variations actually come from inter-rater disagreement. And then our genetic prediction models actually perform well with meaningful prediction accuracy out of fold about 0.16 to 0.22. And these may seem modest at first glance, but actually you need to consider this like maximum prediction accuracy is actually bounded by the low reliability of each single rating. And our models also outperform skin tone polygenic scores, and using this predicted skin tone, we're able to observe steeper gradients than the interviewer rated skin tone. So it's about 3 to 4 times larger for childhood SES and 3 times larger for racial classification. And when we apply these like a prediction model to HRS datasets and compare that with AdHealth, we show that in both datasets, darker predicted skin is always related to fewer years of education. And the gradients are steeper in HRS, so basically suggesting this weakening but also persistent skin tone stratification across cohorts. So the main takeaway is that single interviewer ratings are noisy and likely underestimate skin tone stratification. And the DNA-based prediction measures, they help recover a more stable, portable measure component of perceived skin tone, reveals stronger within race gradients, and also enable skin tone research in datasets without direct measures. And I won't have time to discuss this in depth, but I also want to note here that like this measure can also open opportunities for causal analysis of skin tone stratification. I'm happy to talk about this more during the poster. So thank you so much, and please come by my poster, it's here.
Okay, so last but not least, my name is Weilong Zhang, and I'm working at University of Cambridge. So today I'm going to talk to you about like the revisiting this fetal original hypothesis. Sorry, my title was not there, so I will tell you the intuition by words. So we want to think about the fetal original hypothesis, and this thing is already illustrated by some previous papers. So just talking about what is like the importance of the first 1,000 days and see its long-term consequences in your lifetime. Okay, so usually all these kind of studies was looking at the downstream outcomes, such as the health disease outcome or the economic outcome. So we try to extend this literature in 2 dimensions. So the first dimension is like we want to looking at some kind of more upward upstream outcome, which is like this epigenetic linked aging measures. But just to be accurate, we are using the telomere length measure. So this will be a common measure for the cellular aging measure. Okay, so and secondly, instead of only looking at the average fat, we are trying to distinguish people by their genetic heterogeneities. So here we are using this so-called HLA, the human nucleotide antigen and heterozygosity. So this is actually a common measure about your immune system heterogeneities. So we are thinking about someone is more immune, like the immune system is more resistant than other individuals. So our leading story is like we are looking at one particular initial shock, which will be the sugar shock happened in the UK. I will tell you more details there. But the idea is like when people have overconsumption on sugars, this will lead to the inflammatory shock and the oxidative stress on the cell on cells, right? And this will actually create some kind of telomere length attrition acceleration, and this will create like the biological aging acceleration. Okay, so this is kind of our story. But on top of that, we also want to look at whether such kind of effect will be heterogeneous when people have different immune system. Because the idea is like if someone the immune system is very resilient, then they will be very effectively neutralize all these initial sugar shocks. So they will less suffered of the shock in terms of their telomere length attrition. Okay, so in terms of the design, our idea is like leveraging the derationing of the sugar derationing happened in the UK back in 1953. So the idea is after Second World War, in 1953, like the UK government decided to lifting the sugar rationing. So the immediate idea is like previously, because sugar is completely rationed, so people can only consume up to 40 grams per day in terms of sugar and sweets. But afterwards, you will see like a clear jump up, almost double in terms of their sugar consumption. Okay, so we are kind of doing the cohort comparison, comparing the people who born before 1953. So they were the treatment group in this low sugar environment, and then comparing with the people, the kids born after 1953. So this will be the ones born after the rationing was lifted. So they were starting from this high sugar environment to start with. So this early exposure, we are looking at what will happen in their lifetime consequences, and we are trying to look at the G by E interactions, because not only the environment is changing, like they have different sugar exposure in their early days, but also they will have different like initial genetic endowments. So we are talking about the ones whose immune system is more resilient compared with whose immune system is more vulnerable. So this is where this G by E interaction terms appeared here in our interaction terms. Okay, so in terms of our main results, so I want to show you a few things. First of all, on average, we actually see that the sugar shock essentially will cause the acceleration of the cellular aging. But here you should read this in a reversed way, because those are the individuals born before the sugar rationing lifting. So they will be the ones who had this treatment and exposed to this low sugar environment. And then those ones are born after sugar rationing was lifted. So they will just be the control group who were starting this kind of high sugar environment to start with. But the idea here is we kind of see on average, you see a very significant effect when people are initially born in this low sugar environment. Then this will help them to protect from this sugar exposure, and then their age acceleration can be slowed down compared with the other group, right? But the more interesting idea is like when we are further distinguish people by their immune system, comparing those ones low HLA means like they have low HLA heterozygosity, so they have like a more vulnerable immune system compared with the ones whose immune system is more resilient. Then the resilient group actually had very little impact from this excessive sugar exposure, right? So all the effect was contributed by this more vulnerable group. Then convert these numbers into some kind of an age effect. Then this effect essentially means like everything else equal, because of this early exposure, people on average had like 1.8 years older in terms of their cellular aging. Okay, so they will feel like this will be some kind of meaningful result when you think about this fetal origin hypothesis, and this effect is actually concentrated on the individual with this low HLA. Yes, that's everything. I might post it here. Happy to talk to you. Yeah, later on. Thank you.
So we're gonna start again. This session's gonna well, I'm Sylvia Barcelos. I'm a associate professor here at Population Health Science and the Follette School of Public Affairs. And this session is gonna follow the same format as the session before lunch. So flash talks, it's gonna be 5-minute talks, followed by some time for us to interact with the speakers at the posters. And we have a very interesting multidisciplinary group here coming up. And without further ado, I'm gonna. So who's first? Oh shoot, you're first.
So hi everyone, my name is Shu, and I'm currently a master student in Wisconsin-Madison. So today I'm going to introduce our work quantifying genetic effect heterogeneity across ancestral populations, which is led by Q. And my teammates Yu Chang, Huajun, Yiru, Jiacheng, they all contribute a lot to this project. So next I will start from a very simple example, the ApoE. So in Alzheimer's research, we all know that the ApoE is a critical gene. However, there's differences in its impact between populations. So based on this picture, we can say that the risk of the ApoE44 genotype is over 20 in East Asian populations. However, it is significantly lower in non-Hispanic white, non-Hispanic black, and Hispanic populations. And the ApoE34, ApoE24 have a similar pattern. So this brings up a fundamental scientific question: is this due to the true differences of causal effect, or is it just a statistical bias due to due to the difference of a genetic structure between populations. So our goal is to develop a reliable method to quantify the difference of causal effects, to quantify the genetic effect heterogeneity without bias. So next, I will introduce our method, XPGene. So for our method, we develop a random effect model, just like the the equation, and we treat the population as an environment factor. And we decompose the phenotype into 3 main parts. The first one is the genetic main effect beta G, and second one is the population main effect beta E, and most importantly is that is the gene by population interaction effect beta I. So the beta I is the one we care about most. And in addition, the XPGene doesn't require individual level data. We only use GWAS summary statistics from 2 populations. And then it can give us 3 estimates, the sigma G square, sigma I square, and the rho GI. So basically the sigma I square can quantify the the genetic effect of heterogeneity. So here is a very brief workflow for our method. So 2 GWAS summary statistics as input, and XPGene can output 3 estimates. So after validating our method through extensive simulation works, we put XPGene to work on real-world data. Basically, we analyze 40 traits across European and East Asian population using public GWAS. So the European is a meta-analysis of the UK Biobank and FinGene, and the East Asian is from the Biobank Japan. So as you can see here, our focus is to estimate the sigma I square because it quantifies the genetic effect of heterogeneity. So we identified significant genetic effect of heterogeneity for many traits like the height, body weight, and the BMI, and etc., while finding no significant heterogeneity for LDL, cholesterol, and many other traits. So in conclusion, XPGene provides a much needed solution for quantified genetic effect heterogeneity across population. It advances our understanding of how genetic association differs between populations. So I believe that it will become an important tool in the future for multi-ancestry genetic studies. So that's all for my report. Thank you.
Hi, I'm Philip. I'm a postdoc at the Max Planck Institute for Demographic Research in Germany. And I will talk about mental health and partnership transitions and the role of genetics. So in family demography, the association between partnership transitions, so union formation, union dissolution, and mental health has been a question for like a very long time. And there are like quite well replicated findings for union dissolution. Like the nice thing is that usually in the year of the dissolution, your mental health declines, but then it kind of bounces back to previous levels. Unfortunately, the same is for union formation. So it increases in the year you find a partner or you marry, and then it bounces back. And there has been lots of research on heterogeneity by, for example, gender, by SES. But mental health or like we measure here depression also has a strong genetic component. So in this paper, we wanted to include this in this analysis. So basically the question that is asked in this paper is, do antidepressant purchasing trajectories before and after partnership transitions differ by the genetic propensity to depression? Here already a little peek into the results. So we find little evidence of genetic moderation around union formation. But we find that especially before a union dissolution, those with a higher genetic propensity for depression are more strongly affected. We're using pretty nice data from Finland, where we have full population register data that can be linked to genotyped survey data. As an indicator for mental health, we use the information on whether a person purchased antidepressants in a year, and we can look at kind of a time range from -5 to +5 years around the union events, and we're using individual fixed effects with PGI interactions. The main results I will show you at the poster. Here I can show you already the gender stratified results. So basically for union formation, as you can see here, we have a pretty solid null finding. And for union dissolution, you see that, so the y-axis is actually the interaction effect, that the only time period where something is like more strongly going on is in the years leading up to the dissolution, which might be the potentially conflictual period. So what is this paper adding to the literature? The contributions are first that we are moving from like static comparisons of partnership states to a more dynamic view on the trajectories around partnership transitions. We show that the genetic vulnerability matters most in the pre-separation period. And so the main takeaway is that genes do not broadly shape mental health responses to all partnership transitions, but they matter most in the potentially conflictual period that is leading up to a union dissolution. And for everything else, you can talk to me at my poster, which is over there.
Hello, my name is Senan. I'm a PhD student at Cornell University, and I'm here to talk to you about the direct and indirect effects of genetics and education. So as part of this analysis, I estimate the average causal effect of the education polygenic index, which is a summary index of genes associated with years of education in the UK biobank population. In this graph, I show the colored lines, which is the raw association, as in OLS, between years of education and later labor market earnings. That's the red labor market earnings and the green for education years. After controlling for imputed parental education polygenic index values, I then estimate the average causal effects, which corresponds to the yellow lines, which are solidly not zero, meaning that there is an average causal effect of the education polygenic index on education years and hourly wages and other income measures. Essentially what this does is estimating average causal effects, replicating main estimates in the economics literature recently. Based on these findings, I operate a causal mediation analysis, decomposing the education polygenic index average causal effects on labor market earnings into an education channel. How this works is by estimating the average effect of the education polygenic index on education years and baking it into a decomposition with a direct genetic effect and the education effect. This kind of analysis also relies on using a causal measure for education returns, and that's denoted by the dotted line between the arrows between education years and later labor market earnings. So how I do this in my setting is by operating a sensitivity analysis, being deliberately agnostic about the values for education returns, but also alluding to the labor economics literature on the plausible range. So essentially how this works is I estimate the red line in the middle is the average total effect, so the effect of the education polygenic index on labor market earnings, and then I decompose it into the corresponding channels, which are the green and blue lines. If you think that the correlational estimates, so raw correlation between education years and wages, represents the causal effect, then you would take the mediation estimates, which are along that dotted line, called the correlational estimate, and you would conclude that most of the education polygenic index effect is going through the education channel. The plausible range from the economics literature is a bit larger than that, up to around 10%. And under those values, you would conclude that everything is going through the indirect education effect. Doing this with the other income measures used in the UK Biobank is saying that the amount of the education polygenic index effect on labor market earnings goes through primarily education years, and the direct residual genetic effect closes towards zero at plausible returns to education values. If you want any further detail about this, feel free to see me at my poster or read my paper online. Thank you.
Hi, everyone. Thanks for having me here. I'm Sally Cole from the Department of Psychiatry at Robert Wood Johnson Medical School at Rutgers University. Today, I'll be sharing work on the influence of spousal substance use disorder on alcohol use disorder during marriage using a dyadic and genetic approach. We know that risk for alcohol use disorder is jointly shaped by social and biological influences across the lifespan. So in adulthood, marriage becomes a particularly important context for understanding AUD. Partners often resemble one another in their drinking behaviors. And in some cases, alcohol problem can spread within couples over time. But while we know a lot about alcohol-specific spousal influences within relationship, kind of the alcohol contagion model, we know much less about cross-substance effect. For example, we know that alcohol use disorder commonly co-occurs with other substance use disorder, but we don't know much yet about whether a spouse cannabis or other drug use disorder may influence risk for alcohol problems during marriage. Beyond a spouse's own substance use behavior, we were also interested in partners' predisposition. Most genetic studies have focused on individuals' own genetic risk without considering the partner's kind of predisposition. But we know that in close relationship, a spouse genotype may also function as part of the environment for the other partner, which raises the possibility of social genetic effect that the spouse genotype may matter as well. So in our study, we use a dyadic and genetically informative framework to understand how spouses may influence AUD risk during marriage. We use data from more than 1,000 couples from the collaborative studies on the genetics of alcoholism, which is a large family-based study on substance use and related disorder in the U.S. And we examine both behavioral and genetic influences within couples. So a major strength of using this data set is that we were able to link both partners to have both behavioral and genetic data from both partners. So we focus on two main research questions in this. First, do a spouse cannabis use or other drug use disorder increase the risk of AUD during marriage? And second, does a spouse genetic predisposition to externalizing behavior predict alcohol problem in their spouse, even after accounting for the individual's own genetic risk? So the reason that we use externalizing polygenic score here rather than alcohol-specific polygenic score is to capture the broader genetic liability toward substance use in general. Methodologically, we use a dyadic mixed effect models, which allow us to account for interdependence between partners, and so that we were able to separate out partners' effect from their own individual effects. So what do we find? So first, partners matter strongly at a behavioral level. Having a spouse with AUD was the strongest predictor for alcohol problems during marriage. But importantly, what we also found a cross-disorder effect. So a partner's other drug use disorder that's inclusive of cocaine, opioid, stimulant, and sedative disorder was also associated with increased risk of alcohol problem during marriage. And second, we found evidence consistent with social genetic effect. A partner's genetic externalizing polygenic score was associated with the focal individual's alcohol risk during marriage, even after accounting for the individual's own genetic risk. So in other words, a partner's genetic predisposition kind of may help the relationship environment in ways that influence alcohol problems. So I think broadly our study suggests that partners matter both phenotypically and behaviorally, and then when we think about substance use outcome, we also need to think about the relationship context as well. And our poster is over there, and I look forward to meeting you and discussing further. Thank you.
We were interested in the causal effect of educational attainment on mental health outcomes, but also in the kind of broad methodological question as to whether Mendelian randomization performs well for these complex, socially patterned exposures. And so, just to refresh your memory, Mendelian randomization uses genetic variants that predict your exposure X as instruments for that exposure in order to estimate some causal effect of interest, so the effect of X on Y. And if certain assumptions are met, this can overcome problems of reverse causation or unmeasured confounding at the phenotypic level. But the problem remains that complex and especially non-biological exposures like education can have heterogeneous effects, which can indicate genetic confounding from horizontal pleiotropy. And so to address this, we used a clustered Mendelian randomization method where we use different clusters of genetic instruments. So for example, here, G1 and G2 as instruments for our exposure X in order to then estimate distinct causal effects on Y. And if we see heterogeneous cluster estimates here, that can indicate genetic confounding. So previous studies have found a generally protective effect of education in terms of years of schooling on mental health outcomes, but the mechanisms are not always well understood. And so we did find overall a protective effect of education, but we also found that this seems to be driven by cognitive factors. So this is a bit complicated, but I'll just draw your attention first on the left to our cluster results here. And this is a simplified kind of heat map. The clusters down here are kind of these mixed bags of behavioral and mental health traits, which is kind of consistent with a very polygenic architecture. We also see a physical cluster here in the middle that is enriched by BMI and height. And I would call your attention to this cognitive cluster up here that is enriched particularly by intelligence, executive function, and reaction time. And so on the right here, we see all of our estimates with our kind of cluster, our cognitive cluster in red and all the other clusters in these other colors. And so on these mental health outcomes, ADHD, anxiety, autism, MDD, and PTSD, we see quite consistently that there is an overall protective effect of education, but also that the cognitive estimates here in red are consistently have an outlier effect. And also the Q statistic here underneath each outcome indicates that these estimates, these clusters are statistically heterogeneous. And for autism, a particularly interesting example here, we even see that the effect of the cognitive cluster is opposite in sign to the other clusters, meaning that the other clusters are risk increasing, but the cognitive cluster remains protective. The univariate estimates that we see here, which are the crosses, which are essentially what you would estimate in a conventional Mendelian randomization analysis, which is essentially just an average of all these other instruments, we see will fail to detect this kind of heterogeneity that can be quite easily picked up using clustered instruments. So to put that into context, so here are some control outcomes. So for our positive control, we know that education increases income, and in line with that, of course, we see that all of our cluster estimates are positive, but also importantly, the Q statistic is not heterogeneous. And conversely, we, as a negative control, we know that education has no effect on whether or not you're right-handed. Once again, these estimates are, in line with our expectations. They are null, but again, importantly, we see no evidence for heterogeneity. And so what that means for the kind of findings that I showed you earlier on mental health outcomes is that the heterogeneity that we observe there is likely due to genetic confounding that you can see here on this DAG below. Essentially, horizontal pleiotropy has created a backdoor causal pathway running from cognition to mental health. So in conclusion, the protective effect of education on mental health is driven by cognitive factors. And we also see that clustered Mendelian randomization methods can inform us about the presence and nature of genetic confounding with complex exposures.
Hello, I'm Wesley, a second-year PhD student in sociology at Purdue University, and it's been a pleasure to be back here for TAGC. Thank you for the honor. And today I'll be talking about a project that is currently ongoing and quite preliminary with two of my mentors, Evelina Akimova and Robin Stryker, where we basically want to ask the question, to what extent does EA educational attainment PGI predict educational and occupational outcomes? And we sort of look at this, from a direct effect perspective, where we say, net of all the mediators, how much of this relationship between EA PGI and education and occupational outcome remains. And we also look at it from an indirect effects perspective, where we are wondering, how much of this relationship sort of flows through, social mediators that are informed by status attainment processes. And this research basically builds on a line of other studies that looks at status attainment processes, where we sort of say that, socioeconomic outcomes are sort of the products of not only individual attributes and abilities, but also their aspirations as well as those of the people around them. So this actually is also one of our co-authors, Robin, actually look at the effects of religion and ethnicity and how it flows through the status attainment the processes. And we want to sort of introduce genetics into it by asking, do genetic correlates of educational attainment sort of flow through this kind of status attainment processes that we have looked at so far in classical status attainment models. And so I think this fits very well into the classical, nature and nurture debate. And how we do this is that we use the Wisconsin longitudinal study that I'm sure everybody here is very familiar with, which sort of follows high school graduates across the life course. Our sample size is about, analytical sample is about 9,000 genotypes individuals, and for the sake of this study, we only looked at those of European ancestry. Our key variables are obviously the EAPGI informed by Okbe et al.'s 2022 GWAS, and obviously status attainment variables like their IQ and high school rank to be indicative of one's individual ability, as well as their educational and occupational aspirations. We also look at the influence of people around them, so parental, teachers, and as well as their friends' encouragement to attend college, and a control of a range of family background variables. Our outcome variables are basically those of years of education completed, as well as the first full-time job, Duncan SEI scores. And our analytical strategy is a structured equation model, SEM, that allows us to decompose the effects into direct and indirect effects. And we also then pooled it, and then we conduct a sex stratified analysis to see if this relationship differs by males versus females. So this is just a teaser of the results for the pooled sample. At the top there in the green bars, you will see the bivariate correlations between the EAPGI as well as both of our outcomes, educational attainment and first job SEI. These are quite substantial in amount, but in our second model in blue, we include the family controls, and you see that the effects drop by approximately 24.6% thereof. And in the final model, where we include for all the significant status attainment mediators, we see that there is a substantial drop in educational attainment by about 78.2% and about 37%-ish thereof for first job SEI. And I'll be very happy to talk about this. And like I said, this is a very preliminary sort of ongoing research, so any feedback would be very much appreciated. Thank you, and I'll see you in my poster, which is the second one from the right over there. Thank you very much.
Thanks for coming back. So my name is Tian Yuan. I am an assistant professor here at UW-Madison. I'm really pleased to be moderating this session. So we are in this very exciting field. I'm pretty sure everyone has had great conversation with each other. We know there are more and more genomic data sets. They're continuing to grow both in scale and in diversity. And we have analytical methods that are increasingly sophisticated. There are many outstanding questions we can approach now with unprecedented statistical power and resolution. However, these advances also pose a parallel set of challenges. So in this session, we have a group of talks that approach these challenges from complementary perspectives. So together they will highlight ongoing efforts to develop methods that are not only more powerful, but also more interpretable, more robust, and more broadly applicable across different populations and research settings. So let's get started. So please join me in welcoming Matthew Maxwell from here.
All right. Thank you, T. As you mentioned, I'm from UW-Madison. I'm a graduate student, PhD candidate in the philosophy department, which is a little different from some of the other speakers you'll hear from today and tomorrow. My talk is on population substructure, which some of you may know is going to be an important topic for controlling. If you're doing a case control study, you need to know whether you have multiple subpopulations that might confound some of your data. So I'm going to be talking about structure-like algorithms and the search for disease-linked loci. So as you probably know, population structure and associated linkage disequilibrium can cause spurious correlations. And case control studies, as you also, I'm sure, are well aware, are studies where you have two groups. You're comparing two groups, one with the trait of interest and one with without that trait of interest. And you want to see what differs between those two groups. But like I said, these can be confounded by linkage disequilibrium or other correlations between the population and the trait of interest. So Knoller et al. 1988 is kind of a classic example of this. They find a link between certain GM haplotypes and non-insulin dependent diabetes in two Native American populations, two closely related Native American populations. But this association disappears once you control for European ancestry. And it turns out that these haplotypes are a proxy for population membership. So they're picking up something that they didn't mean to, and they're getting a bad result. Now, this study did acknowledge this and came to the right conclusions. But it is a risk that we face. So the problem is, and I need to do a little bit, this is a different audience than I was first expecting. So the problem is we need to determine what these subpopulations are. And I noticed in a lot of your studies or data sets, what you're looking at are self-reports of population. So you might be looking at race or ancestry. Quite a few people in population genetics are interested in using algorithms to discover those population substructures. So the most popular of these are structure and a similar one that I'll talk about called admixture. And these are used to discover this population substructure. But these have been really strongly criticized, that especially from philosophers and some people even within the population genetics community. So in this talk, what I'm going to do is examine three what I take to be the main arguments against the use of structure-like algorithms in particular human populations. So first, we have that the number of population subdivisions, or K, is an input, not an output of the structure-like algorithms. So when you are getting results, you might get a result and say, there are this many population subdivisions, but it turns out that's kind of arbitrary because you put K in the first place. It wasn't an output. Second, quite a few people have criticized structure-like algorithms as being merely exploratory rather than confirmatory. So the worry there is that you can't actually make any inferences from this. You're just restructuring the data in some way such that you can see some patterns. You're not really getting any meaningful conclusions out of it. Third, people have said there are no objective subdivisions in the human population. And if there aren't any objective subpopulations in the human population, then you're going to end up with a bunch of false results. So I take it that 1 and 2 are objections to structure-like algorithms generally. And then the third one objects to their use in the human population. And I'm going to take all of these on. None of these objections, I think, undermines the use of these algorithms for the purpose of finding disease-linked loci. So I take it that, well, many of these objections have really been raised in the context of racial realism, the idea that there is some biological reality to race, and people have been really keen to criticize that. I'm not going to weigh in on that question of whether there are biological races, but I take it that even if these algorithms do not give us good information about that, they can still be used for this purpose. So what are these structure-like algorithms? I don't know how many of you have run across these. They're pretty common in evolutionary studies. I think they're less common in health and policy-related things like many of you are working on. The idea is these are algorithms that use population genetic data to infer population structure. And by population substructure, I just mean the presence of systematic differences in allele frequencies in a population. And this is often due to non-random mating and population history. Much of this is going to be cashed out in terms of linkage disequilibrium, which I'll have more to say about. So the main two structure-like algorithms are, the name comes from structure, from Pritchard et al. 2000, and then a very similar algorithm, admixture, from Alexander et al. 2009. How do they work? Basically what they do is they have a model-based assumption of what an ideal subpopulation is going to look like. And that ideal subpopulation is going to be at Hardy-Weinberg equilibrium. So that's going to be a particular ratio of homozygotes and heterozygotes in the population. And that's going to have low linkage disequilibrium or low connection between alleles within the genome, even if they're closely spaced. So the algorithm then uses a Monte Carlo Markov chain method to discover the sorting of individuals into K groups that minimizes deviation from these modeling assumptions. Now, structure uses a Bayesian method that assumes a prior distribution. And then admixture just uses a likelihood approach that gets rid of that prior distribution. It's less resource intensive, and the authors show that it provides basically the same results. So empirically similar accuracy. So most people have switched over to using admixture today. Now there's a little bit of a wrinkle. Both of these algorithms don't just tell you just bin each individual into a certain subpopulation. They also give you some measure of like, maybe this individual has partial ancestry from different subpopulations, but that's not really important for today. Now, let's go on to the first objection, that K is an input, not an output. So Weiss and Fullerton, DeSalle and Tattersall, Griesmer and Berrigan argue that K is an input, not an output. So what you end up with is basically when you infer that there are K subpopulations, you're not really inferring anything. That was something that you put in the first place. Most, I guess, well-known is the inference that there are 5 or maybe 6 continental populations. This comes out of the name is gone from me. Well-known 2002 study that I'll cite. And people have really criticized this on the basis that, people have taken this to mean that there are these 5 populations that correspond to races and so on. So these authors have said, well, either K is arbitrarily chosen, you just chose it and put it in there, or you had some preconceived notions of how many groups there should be, and then you put that in there and then sorted. This is kind of correct. K is an input, not an output of the algorithm. But the choice isn't arbitrary, as these critics have claimed. So this is Rosenberg. That's the name I was trying to think of. Thank you. So Rosenberg et al. 2002 compare actually a bunch of different K values and then evaluate them. You might be familiar with this. You might have seen this before. This is the output of the structure algorithm. And you can see the different sort of subpopulations that it comes out with at different K values from 2 to 6. So they don't just run it for one, they run it for a bunch of them. And they compare those against each other. And they have some different ways of doing this. So Rosenberg et al. 2002, they suggest choosing the K which maximizes the log posterior probability of the data while not generating multiple solutions. So basically it's the highest K value that doesn't start to generate these multiple solutions. And there's a problem with this. Higher K generally improves the posterior, so you're not really getting a great result from this. That's been criticized pretty roundly, but they've put some thought into it. They're not just choosing randomly. They have some other possibilities for finding the K value, but they reject them as too computationally intensive. So they don't use those. And ultimately, Rosenberg et al. Doesn't even pick a K value. They're not, so people criticize them for saying that K equals 5 or K equals 6 is the correct number. They don't claim that. It was other people inferring from that and making these arguments for the continental populations from that. Second, there are other ways of doing this, though. Second, Evano et al. 2005 develop an ad hoc statistic. So they're not really giving an a priori sort of justification for this. They base it on the second order rate of change in the log probability of the data. So basically you're going to get improvement as K increases, but what's the rate of change in that? And they show that empirical evaluation confirms the reliability of this method. So you're getting greater, or you're basically, if you model, if you do simulations, this is going to usually pick out the correct number of values or number for K. So then Hulsenbeck and Alfado add a prior probability for K and choose the K that maximizes the posterior. With sufficient data, they argue this swamps the priors. I think there are reasons to be concerned about this, but basically the idea is they're not just picking this out of thin air. Now, November 2016, and even Pritchard in his 2000, do note that these methods still aren't very reliable. Inferring K is still a problem, but it's not arbitrary. Second, we have the objection that structure-like algorithms are exploratory. The idea here is that these exploratory studies are just coming up with, just from data they're trying to show to prove a hypothesis without any, or trying to present a hypothesis without building in the assumptions. They're not testing a hypothesis, they're creating one. And you can't just create one from data, you need to be testing it deductively. I think there are reasons to criticize this distinction, but the real worry is that an exploratory study is always going to return a positive result. So I'm sure you're well aware of PCA studies. This criticism is kind of historically linked to these studies that you have findings of G, the general intelligence factor, P, the general psychopathology factor, and even in population substructure, like this plot from November et al. 2008. This is the population of Europe, and then they're finding the two principal components, and then they note, oh, hey, it's interesting that the Iberian Peninsula and the Italian Peninsula kind of show up. So they infer that we have this link between geography and population substructure. the objection is that it's mathematically necessary that you're going to have these population substructures. And so whether there's a reality to it or not, you're going to get it. So it's not necessary that there's a real world correlate. Now, this isn't so much a problem. So the question is, are structure-like algorithms like this, as some of these people have claimed? In some sense, yes. Given a k, the algorithm is going to return a best fit sorting of the individuals. Now, there's often going to be disagreement between runs, but it's going to give you an output. It's going to tell you that one of them is the best fit sorting. It'll frequently find a best k if a k criterion is used. But on the other hand, no. This is different from PCA. The model maximizes, or the model assumptions of the algorithm interpret the output as subpopulations with two features, namely maximized Hardy-Weinberg equilibrium and minimized linkage. So these algorithms test the possible sortings of individuals into subpopulations, but they do assume there is some structure to be found. This is going to lead me to the final objection, that there aren't any subspecific divisions within the human population. The human subdivisions are all fictional, either because genetic diversity is too low in humans to represent legitimate subpopulations, or the only legitimate subdivisions are clades, but structure-like algorithms don't necessarily find clades. Third, human genetic diversity is better modeled as clines, or constant, rather than having sharp divisions in clusters. But structure-like algorithms are biased in favor of finding clusters. Now, I take it that all the above could be true, but it really ignores the purpose that we're putting this to. We want to control for linkage disequilibrium and other spurious correlations. And structure-like algorithms, by design, subdivide to reduce linkage disequilibrium and avoid spurious correlations. That's exactly what we're looking for. So it doesn't really matter if these things are real. It doesn't matter, even if the human population is clinal, or the subdivisions aren't clades, the goal of reducing spurious correlations is met. And I take it that we can use them relatively safely. All right. Thank you very much.
I just had one. So, if you had asked me, is k an input and output, I would have said, oh no, we use things like AIC or BIC to just we optimize model fit using these sorts of criteria, and that's kind of like the modern way to do things. And I guess that was sort of reflected in and I guess that fits into your discussion here and kind of what the so what is, given that if what we're trying to do is practical and not the research question isn't what is k, but we just want to find a k that allows us to do our research.
Yeah, great question. So Halzenbeck and Lofato both they mention using AIC, but they don't in fact use it. I'm kind of surprised they don't. I don't know why they don't. It might be a computational limit, but they. This is also something my supervisor Elliot Sober is really concerned about. He's like, why don't you just use AIC? Not that I've seen. That's a good question. This is basically what I found, and it seems like people have kind of run with it. So if you know of something more recent, I'd love to hear about it. What's that? Admixture does? Okay. Yeah, we'll talk.
Okay, so in the interest of time, we'll move to the next speaker. So let's thank Matthew again. So our second speaker is Shihui Peng, also from UW-Madison. Actually, all the speakers in the session are from UW-Madison, so welcome.
So hi everyone, my name is Shihui Peng, and I am a first-year PhD student in the UW-Madison. And today I would like to talk about a project about how to toward a more equitable risk thresholding in phylogenetic risk prediction in a multi-ancestral cohort, in a multi-ancestral setting. So let's start with our motivation. So the polygenetic scores are increasingly discussed as clinical tools for stratification because like they can help us with estimation of people's genetic predisposition for multiple outcomes and thereby help with the risk stratification, early disease identification, and also the precision prevention. So why does the polygenetic scores need to work in the multi-ancestral settings? Because in the real world, the populations are highly genetically diverse, and practically the clinical screening requires a broadly applicable tools. So that is to say, the clinical implementation should not rely only on the ancestral-specific tools, and the risk thresholds ideally should be portable across the ancestral populations. So in this case, if we would like a PRS to be clinically and practically useful, this should work equitably well across the ancestors. However, current methods prioritize the predictive performances improvement, such as improving the AUC value and the R-square values in the model, but we didn't find any methods that specifically address the ancestral-dependent polygenetic scores distribution shift. So what is the distribution shifts of the PRS? And here's an example. And the PRS was derived using an European ancestral-only cancer GWAS. So the distribution shift means that the PRS distributions move across the ancestral groups. So if you could notice that the same top 10% thresholds based on the population, it does not correspond to the same percentile across the ancestral groups. So that is to say, some populations are overrepresented and others were underrepresented among those who were labeled as high risk. So this reveals a calibration and equity issue, which is part of what the PRS is capturing may be the ancestral dependent structure rather than the purely disease liability. So this could be a very important issue in the United States because the US has one of the most mixed multi-ancestral populations across the world. So our aim is to account for the PS distribution shift and then to obtain a more equitable and clinically applicable polygenetic risk threshold for the risk stratification in a multi-ancestral setting. So our idea is actually pretty straightforward. So firstly, we reasoned that the PRS distribution was due to the population structures in a multi-ancestral setting. So to be specific, in different ancestral populations, there could be very different allele frequencies across the variants, and also very different linkage disequilibrium patterns. And this could be due to the long-term evolutionary history, migration, or the genetic drift. And this could directly lead to the differences in the PRS distribution. And at the same time, different ancestral populations could have very different disease prevalence because they could have very different lifestyles, or they could be exposed under different environmental factors. So when this is the case, the population structure is actually confounding the relationship between the PRS score and the disease outcomes. And one very straightforward way to mitigate the impact of population structure is to make use of the genetic principal components. So we designed a simple two-stage approach to develop a multi-ancestral PRS. So our idea is to regress out the effect related to the population structures from both PRS side and also the disease trait side. So to be specific, we first regress out the first 10 genetic principal components from each of the ancestral specific PRS, and in this way we could get a set of residualized ancestral PRS. So besides, all these ancestral specific PRS could be obtained using any of the state-of-the-art methods, and we don't really have limitation for that. And similarly, we regress out the same genetic principal components from the trait, and then we can get a residualized trait. And then we model the residualized trait using a linear combination of each of the residualized ancestral-specific PRS and other important covariables, such as the gender and the age. And then we define the linear combination of all ancestral-specific PRS as our meta-PRS. So to test whether this framework can work, we choose the height and short statures as the outcome of interest. And this is because height is one of the most heritable complex traits, with up to 80 to 90% of the phenotypic variation could be due to the genetic factors. And the short statures, we already have the FDA-approved growth hormone therapy for children, so an improvement of the risk stratification could have significant public health implications. So by the way, in the clinical setting, the short statures is usually defined as the height less than 2 standard deviation below the sex-specific mean. And you can see that the clinical definition of the short statures actually doesn't contain any information regarding to the ancestry. So we used the largest height GWAS to date to obtain 5 ancestry-specific PRS weight. And also, they also developed a cross-ancestral weight, which was calculated from a cross-ancestral meta-analysis of multiple ancestral GWAS from the same study. So using this weight, we calculate corresponding scores in the all of us cohort. And when we examine the distribution of these PRS scores, we can actually notice substantial shifts in the PRS distribution. So for example, the European ancestry could be highly right-skewed and the African could be highly right-skewed and the European plot could be highly left-skewed. So this does not mean that one ancestry was genetically shorter or taller than the other one. Instead, it mainly reflects the population structures, as we discussed before, like the systematic differences across ancestral groups. So what would happen if we neglect this shift when we try to predict the short statures? So we can see the two histogram of these two PRS score actually doesn't make sense, because theoretically for those people with a smaller value of the PRS, they should have a higher risk of developing short statures. In this case, the lowest decile of the PRS group, they should have the most of the cases, but that is not what this two histogram was displaying. So still using the all of us cohort, we use our framework to construct the meta PRS. So in the training dataset, we first regress the height and each ancestral PRS using the first 10 genetic principal components, and we obtain the residuals. And we use the linear combination model, which we discussed before, to obtain the beta for each residualized ancestral PRS. And then we move to the testing dataset. We applied the PC coefficient estimated from the training set to get the residualized PRS in the testing dataset, and then we calculated the weighted sum of this residualized PRS using the beta estimated from the training set, and this would be our meta PRS. And then we evaluated the predictive performances of this score and compare it to the other scores. So our purpose is actually not to specifically improve the R-square, but our score indeed can explain the highest variation in height compared to other scores, which is nice. But most importantly, our score can largely mitigate the PRS distribution shift compared to others. So the dashed red line here actually doesn't really have biological or statistical meanings. It's just for better comparison. And we can see that the distribution of the PRS across multiple ancestral groups are much more centered for our score compared to the other groups. And the distribution of the short statures of our score is much more reasonable than the other two, because you can see like the lowest group now have most of the cases, and the overall chain is much more monotonic. So what about other predictive matrix? So here's the sensitivity results if we use the cross-ancestral score to define the risk threshold. The x-axis are the polygenetic score thresholds used for the stratifications. So for example, if someone have a PRS score lower than one of the selected threshold, this guy would be defined as the predictive short stature cases. And all these thresholds were calculating using the whole testing data set, and they are not ancestral specific threshold because we would like to mimic a real world setting when we want to use one single threshold to make the risk stratification in the multi-ancestral cohort. So we can see that if we classified individuals who have the bottom 50% PRS score as the risk group, we would be able to detect 85% of the African ancestral cases. However, under the same threshold, only 0.1% of the European cases, 3% of the East Asian cases, and only 1% of the South Asian cases can be detected. And the performances of other predictive measures, such as the specificity, the precision, and accuracy, they are bad too, because you can see there are a high variation across different ancestral groups. And similar to the cross-ancestral PRS, the European PRS and even the PRS calculated by the PRS-CX method, one of the most well-performed cross-ancestral methods, they both struggle on the predictive performances, as you can notice the high variation. So what about our meta-PRS? We can see that the sensitivity patterns across different ancestries are much more similar now. And 40% of African cases, 41% of the East Asian cases, 58% European cases, and 47% of the mixed American cases can be detected using the same threshold. And the other predictive metrics are also performing well, and they are pretty consistent across different ancestries. So this indicates that our meta-PRS score is a potentially much more generalizable approach for the short stature predictions in the multi-ancestral setting. So to sum up, the existing methods mainly focus on improving the predictive performances in diverse populations, but the PRS distribution shift is still problematic, and this could create difficulties when we want to use one single threshold in the whole population. And we propose a simple approach to mitigate the distribution shift of the PRS, and then we apply this framework on height and short stature prediction, and we found that this framework could lead to a more generalizable and perhaps more equitable polygenetic risk threshold to diverse populations. And thank you for my great mentor, Professor Tia Lu, and thank you very much for listening, and thanks for the support for these institutions.
Hey, so we have time for questions.
Yes, sorry for my ignorance. Maybe this question just reflecting I was not exactly in the literature, but in general, I was just wondering how exactly you can distinguish this part is the nutrition part coming from genetic part, because at the end of the day, like even these two populations share exactly the same genetic distribution, but because their nutrition level could be different, then we are expecting they have different distribution when with like height, for example, and then such kind of differences will I guess we'll enter into the GWAS training you were having. So I'm just wondering, like, okay, how should I really think about your principal components actually capturing the genetic differences rather than the nutrition or general, like the society differences across different groups?
So are you asking about like when we residualize on the both sides, will we remove some signals related to the nutrition part?
Yes.
Okay, so I don't. Why should this be a problem? Even if we remove the.
So put in a different way, when you think about I want to do a cross comparison, essentially the threshold. I'm not sure whether you want to put a generally stable threshold only on the genetic component, or you want to put a general threshold on interaction, also including some kind of environment component there.
Oh, so to answer your question, thanks for the question. So the PS score for here is only for make prediction of whether these people could be a potentially short stature cases or not, and this like they could like when we remove the signals, maybe we could like potentially remove some nutrition related signals, but it doesn't matter because we remove the signal from both the traits and the genetic parts. So the things we want to do is to make multiple ancestry to be more comparable. So it doesn't matter if we really remove some like nutrition related signals or something. What we want to do is if we want to remove these in this group, then we also want to remove the same signal from the other groups. So in this case, we can have a more reliable one threshold for like across the ancestral groups.
Okay, thank you.
Does it answer your question?
Any other questions? If not, we can move to the last speaker. So let's thank the speaker again. So our last speaker for this session is Dr. Yuchang Wu, again, also from UW-Madison.
Okay, good afternoon, everyone. My name is Yuchang Wu. I'm an assistant scientist at UW-Madison. Today, I'd like to talk about a pressing issue of sample overlap between the GWAS cohort and the PGS cohort. So first of all, I'd like to thank my co-authors, Yifei Wu, undergraduate student in our lab, and Yiru Lin, a master's student in our lab who did most of the heavy lifting part of data analysis, and also Professor Qingshu Lu, my PI, for his guidance and supervision. In sample overlap is a known issue in the statistics and machine learning literature. It is well known that you have to the training data and the testing data set should be independent of each other to have meaningful model predictions. And in those fields, it's the best practice is to make sure there's no sample overlap between the training data and the testing data set. In genetics field, a polygenic score, or PRS, is a weighted sum of individuals' allele count, where the weight is trained using the GWAS sample size, GWAS samples. So therefore, to have any valid PGS inference, it requires zero sample overlap between the GWAS dataset and PGS sample dataset. So in our field, we rarely talk about this issue. It is not because we are naive, but because practically it was not an issue in the field, because when the GWAS and the PGS cohort sample size is relatively small, and sometimes even those two cohorts are from different countries, and the sample overlap never exists. However, when in this era of exploded large biobank data sets, such sample overlap between the PGS and the GWAS cohort becomes mathematically unavoidable. For example, the UK's dataset, Our Future Health, aimed to recruit up to 5 million participants, reaching about 7% of the UK population or 10% of adult population. That means that for any other UK-based cohorts, about 10% of their participants also enrolled in the Our Future Health. So make things worse is like such overlap is cryptic because we don't know who are the overlapping samples. And also it is pervasive because such overlap affects the GWAS across different diseases and traits. And also it's substantial. So such a large fraction of the population is now enrolled in these biobanks, making this overlap unavoidable. So first, we want to check exactly how problematic this issue is. So in this plot, we made some simulations. First, I simulated the phenotype using UK biobank samples under very standard settings. And then I ran a GWAS using about 10,000 samples. And then I calculate, using this GWAS sample stats, I calculate the PGS for 50,000 participants with varying degrees of sample overlap from couldn't see the laser, from 1% of sample overlap, or 5%, 10%, all the way up to 90% of sample overlap. And then I ran a regression of phenotype on the PGS to get the regression R-squared and the effect size estimate. I can run this regression using only the non-overlapping samples indicated by this green curve, or using the overlapping samples represented by red, or I can use all the samples represented by the black. Then I repeat this process 50 times, and here the diamond represents the mean values across 50 different repeats. So the first row shows the under the null where the phenotype heritability is 0. As you can see that clearly we see an inflated R-squares. I couldn't see the laser pointer, but you can see inflated R-squares as we increase the sample overlap fraction shown on the x-axis. Because this is under the null, we should expect the true PGS effect size is 0, so we can calculate the type of error rates. You can see that we have substantial type of error rates, even we have only a tiny overlap fractions, and the type error rate can be easily as high as 100%. You may have only 5% of overlapping samples. However, if we look at only the green one, where if we run the regression using only the non-overlapping sample, we do not see such inflation. The second row shows the heritable traits, where I set the heritability at 10%. Similarly, we can see inflate the R squares as we increase the sample overlap fractions, and also upward bias the regression coefficients. So clearly, a sample overlap is highly problematic. So do we have a solution? So the answer is no, unfortunately. So to the best of our knowledge, there are two methods addressing this issue. The first one is called eraser. So the intuition is this: you have a publicly available GWAS thermostats. Then you run another GWAS, same GWAS, but using only your PGS data set. So the intuition is under the null, when the treat heritability is 0, if there is no sample overlap, the genetic correlation between these two GWAS should be 0. So therefore, eraser applied linear combinations of these two GWAS effect size to create two new thermostats to ensure the correlation to be 0. And then based on this uncorrelated thermostat, you can calculate even the PRS even for the overlapping samples. However, we believe this method has fundamental flaws because the newly created effect size estimate is a linear combination of the two original ones. You can show that it has type error inflations. Furthermore, because the entire framework was derived under the null with the heritability of 0, it cannot be directly applied to the case when the heritability is non-zero. And practically, you find that eraser tends to undercorrect the overlapping samples, while overcorrect the non-overlapping samples. As an example, here is showing this plot is there's a sample overlap between two Alzheimer's disease cohort, AGAP and ADNI. So here we calculate the PRS using AGAP thermostats for the ADNI samples, because in ADNI samples there are a large fraction of overlapping samples. If there's no adjustment, you can see a large AUCs when we use the ADPRS to predict the AD status. And then the second column shows the eraser result. You can see that the red one for the overlapping sample, there's not enough correction. However, for the green bar, you can show that there's overcorrection for the non-overlapping samples. So practically, this method does not really work. Another method called OAPRS, or overlap adjusted PRS. The intuition is if you know exactly who are the overlapping samples, you can run the same GWAS using only the overlapping samples, and then you subtract the GWAS thermostats from the public available thermostats. Then the remaining ones has no contribution from the overlapping samples. Therefore, you can use this remaining thermostats to calculate the PRS even for the overlapping samples. So this is essentially the inverse of meta-analysis. You can see that the third column shows that OAPRS works pretty well, but the major limitation is that you need to know exactly who are the overlapping samples, the information we usually do not have in practice. So, okay, maybe practically or ideally, if we know, if we have access to individual level data, we can address this issue. However, we think with large sample sizes, this will create a large computational burden, and also whenever there's new recruitment, you have to update your result to rerun the computation to identify the overlapping samples. And more importantly, nowadays, all this data live in the cloud. Even if you have access, individual level access to each individual cohorts, those cohorts live on different clouds. You cannot simply merge them. So therefore, it is practically infeasible to address this issue. So this is an urgent need without a solution. So here we actually propose two simple solutions to this problem. So the goal here is we want to identify who are the overlapping samples, and then we can simply exclude them from the PRS analysis. So we first look at their distributions here. So for example, we show the distribution of the PGS. Different colors represent the true overlapping status. We find that the overlapping and non-overlapping samples have the distribution have the same means but different variances. So specifically for the PGS distribution, you can find that the overlapping samples are more likely to reside on the PGS tails. If we run a regression of phenotype on PGS to get the residuals, we find that the non-overlapping samples are more likely to reside on the tails of the residual distribution. That means for the overall distribution, it is a mixture of two normal distributions with same mean but different variances. So that means you can apply expectation maximization or EM algorithm to identify these two distributions. The EM can estimate the means and the variances and the proportions of these two distributions. And more importantly, it can also generate the posterior probability of, for each individual, the probability of belonging to the overlapping group. So we can run EM using only the PGS as input. We do not need to have a phenotype level information. Or if we have, we can also use the residual as input for EM, or we can use both the PGS and the residual as input for EM. Then we realize that we do not, although EM is very simple and computationally fast, we do not really need to run the EM. We can simply exclude the samples on the PGS tails because the overlapping samples are more likely to reside on the PGS tails. Later, I can show that this method actually also works pretty well. So then we want to test whether using the PGS or using the EM posterior probabilities can indeed predict the overlapping status. Here shows the result for a variety of combinations. Here the x-axis shows the sample overlap fraction. The y-axis shows the AUCs. Different rows and columns shows different combinations of GWAS sample size and PGS sample size. So several observations. First, the performance of using PGS and the EM posterior are pretty comparable. Second, if you fix on a column, if you change the different PGS sample sizes, there's not much change. So the PGS sample size does not really matter that much. But however, if you fix on the rows, fix the PGS sample size, is we change the GWAS sample size, we see a large differences. For example, when the GWAS sample size is small, for example, it's only 5,000, the AUC can be as high as 90%. As we increase the GWAS sample size to, for example, 300,000, the AUC is only slightly higher than 50%. The intuition is this. The GWAS effect size estimate beta hat is a true beta plus a noise. For the non-overlapping samples, this noise is uncorrelated with the individual's genotype. However, for the overlapping samples, this noise becomes correlated with the genotype. This is the root cause of the problem. With increasing the GWAS sample size, the noise in this beta hat becomes smaller and smaller. Therefore, the problem it creates becomes smaller and smaller. The distributional difference between the overlapping and non-overlapping samples becomes smaller. Therefore, it is difficult for this algorithm to distinguish these two distributions. So since using the PGS, it's a simpler and approach and has similar AUC performances. So we next examined whether trimming out the PGS outliers could be a good, a simple and effective way to address this problem. Here, the x-axis shows the percentiles to be excluded from the PGS tails. The y-axis shows the regression coefficient. Different rows shows different degrees of sample overlap, and different columns shows the different GWAS sample size. The green one shows the result using only the non-overlapping samples. You can see that trimming the PGS outliers does not change the regression coefficient if there's no overlapping samples. The black one shows when there are overlapping samples, you can see that trimming out the outliers can reduce the regression coefficient substantially, especially when the GWAS sample size is small. However, when the GWAS sample size is large, such reduction becomes smaller, especially when the overlap degree is small. However, when there's large overlap, even if we extreme 50% of samples, we can still have a large inflated regression coefficient. So then we next use some real data applications. So we first compared ASPEC versus UK Biobank. From this paper, it is found that among about 50,000 ASPEC women, about 1,000 of them also enrolled in the UK Biobank. This paper found these overlapping samples because they have access to individual level data. They use demographic and unique IDs to identify those overlapping samples. So here, we calculate the PGS using the GWAS thermostats from this paper, which mostly based on ASPEC women. Then this table shows that the EM algorithm produces pretty much comparable estimated overlapping samples with those found in this paper. Then we checked how about the tails on the PGS distribution, because we know those overlapping samples should be females, and also they should come from the Bristol area. So we calculate the proportion of females and the Bristol participants and Bristol females among only the PGS tails, and then compare them with the overall proportion in the entire UKB dataset. We can see a large the proportions in these tails are much higher than the background proportion, and we have a very large enrichment of these proportions. Then we show that our method is so robust that you actually do not need to have a real phenotype. Here we compare WLS versus all of us. Here we simulated the phenotype using the WLS samples, using only those who were born in 1939, and then we ran the GWAS, and then we used the thermostat to calculate PGS for the all of us samples. Again, we check those who were on the PGS tails. We find a large enrichment of Wisconsin participants, those who were born in 1939, and Wisconsin participants who were born in 1939. So showing that our method can be simple and very effective identifying the overlapping samples. So to conclude, the sample overlap is a pricing issue that could cause inflated R-squares and type 1 error inflations and upward biased PGS effect size estimates. And the trimming PGS outlier or EM algorithm can be a simple and effective approaches to address this problem, especially when the GWAS sample size is smaller. So we believe that in the future, trimming out PGS outlier samples should be a common and standard sensitivity analysis in any future PGS analysis. A large reduction in effect size estimate is an indicator of large sample overlap. And also, the one limitation is that so far we cannot completely remove all the overlapping samples. Therefore, the PGS regression results should be interpreted accordingly. That's all. Thank you very much for your attention.
Thank you. So we have some time for questions.
Hi, very interesting. So around like 2009 or so, there were several papers that showed how the availability of summary statistics from GWAS can help determine whether a certain genome is included in the GWAS. So essentially, this is like offering something very similar, right? You have a GWAS, and then you have another genome, and then you would like to tell whether it's in there.
Yes. We only have GWAS summary stats, and then we have apparently individual level data for the PGS cohort. We want to know who are those PGS samples were also enrolled in this GWAS analysis.
Okay. Yeah. So I mean, there's quite large literature. I mean, I think nothing was done since 2015 or so, but there were a lot of papers. But also there is a related question, which is whether this is something ethical to do to determine whether someone was part of a UK Biobank, for example, or any other study. Is that something that you thought about? I mean, because you have your genome, the person who donated the genome to your study, and then you're trying to find out whether he or she participated in another study. And that's information that you're not supposed to have, right?
Yeah, there could be some ethical concerns. Our concern was not to identify those. We are not, want to exactly identify those who are participating in another cohort or study. Our purpose is want to have valid PGS inferences.
Right.
To do that, we have to exclude those who are likely to contribute to other to the GWAS studies. But I agree that they could be potentially some ethical concerns.
Okay. Thank you.
Other questions? I can leverage my privilege as a moderator to ask one. So maybe I have asked this before. So does this method assume normal distribution of the PRS? And if the PRS is not supposed to be normally distributed, would it still work? For example, for the Alzheimer's PRS, if you have ApoE included, so it's not supposed to be normally distributed. So would that still work?
The EM assumes a normal distribution, but for the PRS outliers has no such assumption. But for your specific question, I think what matters really is the signal to noise ratio, or the noise to signal ratio. For the ApoE, the signal is so large, the noise is relatively so small. Then I don't think the sample overlap is a problem. Yeah.
Thank you.
Yeah, I was also wondering, you're doing this pure for PGI prediction, but if people do other things with the PGI, like within family analysis or regressing it on other types of outcomes, to what extent does trimming the outliers still work?
Yeah, that's an interesting question. We haven't explored those, but in general, if you want to have valid PGS, then you should avoid sample overlap. So even for within family analysis, I believe that we should also exclude those samples. Our results show that if there's no sample overlap, trimming the outliers does not change the regression coefficient, as shown by this green.
Okay, thanks.
Okay, so I think this concludes this session. We will have a group photo in the atrium of the building. Maybe we can all be outside in a few minutes. But let's thank the speakers again. Thank you for the session.
All right, well, we have our first keynote speaker. It is my absolute pleasure to introduce Dr. Alison Aiello. Her research explores how economic, psychological, and biological factors shape healthy longevity, cognitive function, and the development of Alzheimer's disease. Her work focuses on how stressful experiences across the life course influence biological aging, particularly immune system aging and immunosenescence, as a key pathway linking stress and health throughout the aging process. She's also Deputy Director of the National Longitudinal Study of Adolescent to Adult Health, or AdHealth, and she led the development of the AdHealth Cognitive Assessment, Physical, and Sensory Function Protocol, or ADCAPS. In addition, she has also directed the collection of physical and sensory function measures, importantly, as early indicators of cognitive decline. And one thing that's not even in this bio is that she also led the collection of epigenetic data in AdHealth, which we were just talking about at lunch, is going to be an incredibly important sample for studying aging because it's this rare midlife sample. So stay tuned for a lot of really interesting work coming out of AdHealth. And she's going to share some of that with us today, I think. So we're excited to hear about integrating social, immune, and epigenetic drivers of healthy aging. So please help me welcome Dr. Alison Aiello.
Thank you so much, Lauren. I really appreciate it. And thank you, Jason, for the invitation to be here today. It's really great. And good afternoon to all of you. What a great day today. We've had equal parts methodological rigor, a lot of social science creativity, and excellent collective efforts on adjusting for confounding. So my talk today is actually going to hit on some of those things. I'll mostly be talking about sort of some of the implications for epigenetics research, but I'm also very interested in thinking about genetics and the immune system too, so would be happy to discuss that too as it's of interest. I know we've had some presentations today that have discussed genetics and immunity. So my main points for discussion today are going to be talking about the immune system as really an integral part of healthy aging. And whenever we're looking at a health outcome, many that we talked about today, immunity is related to many, many outcomes. And so we're going to be talking about that. I want to just kind of give a plug for immunity and thinking about immunity. And in much of the research that we do, it may not always be front of mind, but it's really implicitly part of epigenetic research. And I'll talk a bit about that too. And then I also wanted to mention that I think we're entering a new frontier for really understanding immunity and aging. And that very much relates to pioneering work that's being done in genomics today. And I think that's important as a social scientist too, because if we really want to understand how social factors get into the body, then we need to be able to kind of map out these processes that occur, immunity being one of the most important processes for our body and keeping us healthy. Okay, so my agenda for today. The first part, I'm going to just talk about a little introduction to immune system aging. Some of you may have a very strong background in immunity, so maybe this won't be anything new to you, but I just want to kind of set the stage. And then the second part, I want to talk about why we should care about immune aging, especially as it relates to epigenetics. And then I want to talk a little bit about the social environment and how that might shape immune aging and kind of where we're going from here in our understanding. So, when we think about immunity, it's a major component of many of the sort of factors that change over time as we age. So in older adults, we can think about things like reduced muscle mass, which is related to sarcopenia, for example, and skin atrophy and reduced function of mucosa that goes on, and changes in genomic changes also that are happening in a genetic background. But a major key aspect of that is also reduced immune responsiveness, also known as immune aging and also called immunosenescence. And so there's many factors that influence many of these intrinsic host factors. So we think of things like diet and physical activity, frailty, stressors, especially psychosocial stressors and social factors, pharmacological treatments, clinical kinds of conditions too could all influence aging and this immune responsiveness. So there's two different arms of the immune system. We have our adaptive, which is really our specialized arm, and then we have our innate, which is considered more of the general. And for the innate, we think about kind of that first barrier of defense against foreign bodies or injuries and pathogens. So it could be bacterial killing substances in our mucosa. It could be protection on the outside of the skin, for example, our microbiome. But really, this is that sort of first line of attack that our body mounts when we encounter an antigen. Then there's the adaptive component, and this is more specialized. It takes more time. So this is usually against a specific pathogen or body cells that are kind of happening in our body over time after we are exposed to an antigen. So for example, we might have our B lymphocytes may change and counteract an antigen. We may have antibody responses that are going on that our cells elicit. And we may have defense in our cells that are occurring in relationship to our T lymphocytes. And so I'm going to really focus on the adaptive arm today and talking about these pathways because the adaptive arm appears to be very, very important in immunosenescence. That's not to say that the innate part is also important, but there's some aspects of the adaptive part that we can actually map out molecularly now, and where we could not do that before. So I'm going to be talking more about that. So what are some of the adaptive cellular components of the immune system that help us understand immune aging? Well, the thymus, that's a really important one. It's actually been in the news recently, and it's interesting because there's been research on the thymus for a long time, many decades, but it pops up every now and then as an organ of interest for researchers, and there was recently a study showing that the thymus was really important in cancer, for example. And so when individuals are born, they have a really huge thymus. It's one of the largest organs, and it sort of sits right there in the middle of the chest, you can see there. But after 1 year of age, it starts to shrink. And then in adolescence, it drastically shrinks, okay? And so by the time a person is older, their thymus is very small. And traditionally, when cardiovascular surgeons were going in and trying to do work on the heart, surgery on the heart, often they would actually just remove the thymus. So this was happening kind of traditionally. It was thought that as people age, the thymus is so small, it's no longer important, and they would just remove it during surgery. It turns out the thymus is actually important as we age. We do need these thymocytes that are emitted from the thymus. And so what it does is it really emits novel T cells, and that's what's important. But as you age, you have fewer and fewer novel T cells, and I'll talk a little bit about that. But we can actually measure thymic function molecularly. So you can use something called the ratio of signal joint T cell receptor excision circles, and these are known as SJ-TREX. So it can be analyzed by PCR and genomic DNA. And it allows you to actually estimate the number of naive T cells from the thymus that are being emitted from the thymus. So we actually did this a number of years ago. We measured thymic function in DNA in our Detroit Neighborhood Health Study. This was a cohort study of individuals living in Detroit. And what you see here is what you would expect, that individuals who are around 18 to 34 years of age have much higher levels of mean thymic function, meaning that they are producing a lot more naive T cells compared to people who are 65 years and older. And you see some differences by males and females in thymic functioning too, where males actually appear in some of the younger ages, at least in our data, to have a little bit of stronger, higher level of mean thymic function. But as they get older, they have less thymic function. So there's kind of a flip that occurs between males and females. But there's also, once those thymocytes come out of the thymus, you have these T cells are novel. What happens is they begin to age. So you start off with these naive T cells, and there are certain cell surface markers that you can measure. And usually, traditionally, this has been done by flow cytometry. So, for example, you can see this first kind of true naive cell here where it has certain markers like CD28, and then they undergo this aging that occurs over time, right? So they're emitted, and then they encounter pathogens, they become aged. And they become central memory cells, and they become effector memory cells, and finally, at their most exhaustive state, they become terminal effector memory cells. And you can see here in this figure with all the dots there, where people who are 20 to 30 years of age, they have a much higher proportion of naive cells compared to their effector cells, which are sort of the larger cells there, the older aged cells. And then by the time they are 75 years and older, most of their cells are effector memory cells. Are these older sort of senescent cells. They have far fewer naive cells. And that's why, sometimes you may hear, if you give a person a vaccine in older age, they may not have as much of a robust immune response because these changes are going on in the background in terms of individuals' immunity as they age. So there's many markers of immunity that change with aging. So as I mentioned, the thymus, but also the bone marrow decreases over time. We may have a decrease in our naive T and B cells, phagocytosis, so sort of ability to take up antigens, and chemotaxis changes over time and decreases. Antibody production decreases. And again, that's why with a vaccine, you might have to have an adjuvant in older age. Cytokines, though, on the other hand, increase. So you may have heard of this idea of inflammaging. You get higher inflammation as people age. The myeloid cell numbers also increase, and your dysfunctional memory cells also increase with age. And as I mentioned, the vaccine immune response gets worse over time. So these are just some of the changes, but it's interesting to note, too, we often think of there's a decline in immune function that occurs over time. Yes, some things do decline, but some things actually increase over time. And it's not as though having those kind of effector memory cells is necessarily a bad thing, because they do provide protection against pathogens that people might have been exposed to at very young ages. So if they're exposed again, so for example, like the flu, there have been flu pandemics where older people were more protected because they had been exposed to those strains in early life. So there's been a lot of talk about actually hacking the immune system for longevity. And so there's lots of this work that's being done, mostly in mouse models. So some of the ideas are, can we implant immune cells from the spleens of young, healthy mice into older mice and make them reverse aging? Can we remove senescent cells, those TEMRA cells that I mentioned, or block inflammation? Can we regenerate the thymus? And there's been a focus on epigenetic reprogramming of the immune system. But we still don't know how to precisely measure immune aging molecularly. We're still not exactly there. So I think some of these things are in kind of early stages, but they're not. It's not really at the level where we can study some of these things in humans. So why should we care about immune aging when it comes to epigenetics in particular? Well, we know that every cell type has an epigenetic identity. So when we're talking about cells, what we're really saying is immunity. So cell type effects may actually be larger than the exposure you're studying. So when we're thinking about some of these social factors and we're looking at them in relationship to epigenetics, you may actually be sort of swamped by some of these immune components. And that's really important to think about. So in epigenetic research, the thinking goes, if you don't control for cell type, you can mistake what we would say is ordinary biological differences for a disease, treatment, or environmental effect. Okay, so that's sort of how the thinking goes here often in epigenetics. So what's the real concern here? Why are people worried about, immunity in epigenetics or controlling for these cells? Well, this is an example. You could, for example, have a group of individuals in your study, and that group may be exposed to something, and they might have higher neutrophil levels compared to their lymphocyte levels. And these are going to be cell mixtures that you're going to get when you test for epigenetics. Or you may have another group that has higher lymphocytes compared to the neutrophils, and this can skew things when you actually do your epigenetic testing. So early studies in rheumatoid arthritis, especially the EWASs that were done on rheumatoid arthritis, they actually found a number of sites, epigenetic sites, that distinguished rheumatoid cases versus controls. But what happened was that this actually, these identification of all these methylation sites helped actually elucidate the issue related to immune cells and the control of immune cells. So this is a figure from Liu in a paper that was published in 2013, where they looked at methylation differences in cases of RA versus controls. And in A, you have the sort of the methylation differences without adjustment of the cells. And in B, you have it with estimated cell proportions adjusted. And what they found was that a majority of those reported differential methylation cytosines were actually accounted for by an increase in the neutrophil to lymphocyte ratio. So this was sort of the light bulb moment. Okay, we need to be controlling for cells when we ask these questions related to epigenetics. And so you have papers by Lowe, for example, that talk about correcting for cell type composition and the bias related to it. You have Teschendorf, who's done a number of papers talking about cell type deconvolution and epigenome-wide association studies, a review and recommendations of how we should really approach this issue. So what are we told to do about it? Well, we are told that we should adjust for estimated cell proportions. So for example, this is looking at GRIM, just take an example like GRIM H2 and education. So a social factor, looking at biological aging as an outcome. What you might do in your regression is you might add immune cell composition into that regression, adjusting for other factors as well. So the best practice for this lately has been sensitivity analysis. Okay, we're going to put the cells into our regression. We're also going to run it without it, and then we're going to see what happens. So if you have room in your paper, usually it's a brief statement or quick discussion of the role of cell proportions. Often the word immunity is not even anywhere to be found in some of these papers in the association. So and then these differences, the sensitivity analysis is often relegated to the supplement. But I would argue that this is actually really important. And I think we need to be talking about the immune system more. And I say that because the social environment has a very big impact on our immune system, and especially immune aging. So when we think about the social environment, how does it get into our body to affect our immune system? I'm sure many of you have said sort of like, I was stressed out and I ended up getting sick. We see that happen over and over again, just kind of, people's subjective feeling about it. But there are studies to show that the social environment impacts our central nervous system directly. And that leads to a cascade of hormones that then elicits a number of cellular responses. So glucocorticoid hormones, for example, can affect our T cells, our natural killer cells, our monocytes, our B cells, and then that can lead to increases in inflammation, for example. And it's also cyclical because the inflammation can influence our brain in terms of how we feel and that kind of sickness syndrome that occurs when we actually get sick. So every time that you are controlling for cells in your analysis, you're actually acknowledging immunity. But the question is it a nuisance variable? Or is it a mechanism? Are you actually removing the primary way it gets under the skin? So we can think about something like education, for example. Education has many effects, right? It's a kind of a large social, contextual, quality, many other ways in which we can measure education variable, and it's likely that it influences our immunity. And it could be for very different reasons. It could be because lower education levels increase our levels of stress, and then that affects and maybe alters our immunity. But it could also be that the educational environment that we were in may have led to a greater susceptibility to infectious diseases, right? There's also a network component related to education that could influence immunity. And so those things could be on the pathway to things like DNA methylation aging. So these are just things that I think it's important to think about and maybe extrapolate a little bit more in our studies as we work on epigenetics. And the other thing that I wanted to mention too is that these social stressors really impact T cell surface markers. And so when we measure these cell components, we are not necessarily measuring T cell surface markers. So we have things like the CD4-CD8 ratio, which we can get from some of the cell deconvolution methods. We also have CD4 or CD8 effector to naive ratios. So we actually created this ratio a number of years ago based on some flow cytometry data that we had, where we use cell surface markers from naive T cells and cell surface markers from effector T cells and created this ratio to basically get at whether somebody had a higher proportion of these older T cells versus these younger naive T cells. And so we did this. This is a tiny study. You're all going to laugh with your 300,000 person samples because this is a tiny, tiny study of 79 individuals in which we had flow cytometry data. And we created this ratio measure of their effector to naive ratio using the CD4 T helper subset, the CD8 cytotoxic subset of cells, and then their CD4-CD8 ratio. And all we did was look at whether a 10K decrease in income was associated with this effector to naive ratio. And what we found was in this very small sample, even adjusting for a number of different factors that could influence these associations, for the log CD4 effector to naive ratio, it was equivalent to basically an 8-year increase in your aged cells compared to your naive cells. And for CD4, it wasn't as robust as in the CD8 cells were not as robust as the CD4, but still it was about a 4-year increase in the CD8 effector to naive ratio. And that's quite robust for a tiny sample. So you might ask, well, okay, well, that's a tiny sample. Well, more recently, HRS has actually collected T cell data. They did flow, which is great because now you can look at this at a really, really large level. But, you know, this is an older population compared to our Detroit study. They were an average age of 45. But we looked at education here, given some of the issues related to income in the health and retirement study in older populations. But we see very similar patterns here in that individuals with less than a high school education, for example, have a significantly higher effector to naive T cell ratio for CD4. Same with the CD8. Again, it's not as robust as in the CD4 T helper cells. And then we also, in this older population, do see an effect on their CD8, CD4 ratio too, which we didn't see in the younger population that I showed you earlier. I have a few reasons why I think that may be. I think as individuals age, that ratio changes pretty drastically. So we may just have, you know, it may be a more sensitive population to actually sort of see that change in the ratio of the CD4, CD8 cells. So how do we measure the immune cell variables? Well, flow cytometry is the gold standard, but it's really hard to do. So in our Detroit Neighborhood Health Study, when I started working with the immunologist that I was collaborating with, Graham Paulek, who is in Tübingen in Germany, he said, okay, you know, I'd be happy to do the flow cytometry work for your study, but you have to collect the cells from individuals, and we were doing a household study, and get them to the lab within 1 hour to process them. Otherwise, you're going to have a bias set, and, you know, we can't analyze the data. So we had to set this study had, you know, over 1,000 individuals. So we had to basically have a courier at every person's house when we went to their house and drive in Detroit from whatever household they were in to Wayne State within an hour. And we had to make sure there was always somebody on call in the lab to analyze those samples. And so it's technically difficult to do in addition to the technical difficulties of actually collecting these samples. I'm amazed HRS was able to do this. It's such a huge accomplishment. I think they were able to validate for a longer period of time that the cells were sort of out in transit because they have people all over the country, but they did it, which is incredible. We also have the epigenetic cell estimators, which can estimate some of the same things that you can pick up in flow cytometry. So you get cell composition. Really, it's just it's not a cell marker. It's just a percent of CD4 or CD8, percent of monocytes. You know, this could be Hausman, for example, that you'd adjust for, percent of granulocytes or percent of natural killer cells. Now we have deconvolution, which has just come on the board. And I have flow here on this grave because I'm thinking is flow going to go to the wayside because we might be able to actually use epigenetics to estimate all of these different cell surface markers and changes that are occurring in individuals as they age. So in this case, this asks, what is the proportional DNA methylation pattern characteristic of differentiation states of cells? So it's basically kind of looking at cells, what can I identify in terms of the cell surface markers, and what proportion do I have within this cell subset that represents these effector memory cells, for example, or naive cells. So this is an emerging area. I've seen a number of papers looking at this DNA methylation-based immune cell aging composition from blood DNA, but it really does offer a nice scalable alternative for population research, particularly. But we don't actually know the validity relative to gold standard at this point. For flow cytometry, kind of remains that gold standard, particularly when we're looking at population-based studies. And So one example is Lucas Salas's work on this deconvolution method that he came up with, and it allowed us to look at a number of different T cells and a number of different cellular markers in whole blood. And what they did was they had some clinical samples and other samples to validate this on, which is, really great. They were able to do this. They had both the T cells and the DNA methylation, and they created these new markers that allow us to actually look at immunosenescence and immune aging through methylation. So we actually used this algorithm. We applied it into the National Longitudinal Study of Adolescent to Adult Health. And here we're interested in immune senescence itself as an outcome. And we looked at social relationships. This was work by my former student, Fariza Robb, who's now part of our research group. And she actually was very interested in sort of social networks, and that's a social factor that we know is important for immunosenescence. We know it's important for immune function, and what she showed was that people with strong social relationships, in particular, had lower levels of effector memory to naive ratios, meaning that they had less aged immune systems. And this was particularly profound among people who reported that they had high quality family member relationships. We saw there was a little bit of signal for having a high quality relationship with your spouse or your partner, but the family relationships actually mattered the most. And we really didn't see a big impact on having high quality friend relationships. And the kinds of questions that they asked to get at this was like, are you able to open up to this individual and tell them things? Are you able to rely on them? Do they make demands of you or criticize you, for example, which was then reverse coded to kind of create this measure of their social relationship. So the other question that we might have then is, we looked at this in AdHealth. AdHealth doesn't have T cells. Okay, so that's a study where we have the epigenomics, but we don't have the T cells. So we can't really validate it in that study. So the question is, how do these Salas measures actually stack up to flow in population research? Are they actually measuring what we think they are measuring? It did go in the direction that we would expect, but what about the specificity? So because we had flow cytometry in our Detroit Neighborhood Health Study, and we had epigenetic data, we were able to make the comparison. And what we found was actually strong agreement for a number of the cells from their deconvolution method. So the naive and memory CD4 ratios, the percent of CD4 and CD8 naive cells, and the percent of CD8 memory cells had decent agreement. A little bit more moderate agreement for things like the CD4-CD8 ratio and the percent of CD4 memory cells and the naive and memory CD8 ratio. And so it explained around 30 to 50% of the variance for most of the T cell measures. So when we brought this into HRS, which also fortunately has both the T cells as well as epigenetic data, we saw good agreement for natural killer cells that they had, 0.50, but not so great agreement for a number of other cells. So monocytes, for example, naive B cells and memory B cells explained a much smaller proportion of the variance. So there's concerns there. I'm not exactly sure why that's the case. It's unclear. This is an older population too. As I mentioned, our Detroit study is younger. They're age 45. We do, when we sort of spread out the data, we do see some differences in in outliers. Like, so individuals, for example, who might have kind of really low levels or really high levels, there's less agreement. So it's really kind of more in the middle for these individuals. So, to conclude, epigenetics, I think, presents really great new opportunities for understanding how our immune system ages. It also really provides us with a great opportunity to understand how the social environment gets into our body and leads to these immune system aging that we are observing. But I also think we need more population-based research on immune aging. We need to really understand what's happening here. I think we need more validation across these studies in these populations, in representative populations, to understand whether some of these epigenetic markers that we're picking up on and using as new measures to get at, as surrogates, to get at things that are maybe more technically difficult to test, are they actually valid in population-based research? All right, and so I want to say thanks to the IELO research group, my fantastic students who worked on this, and trainees, and our NIH funding, and our Robert N. Butler Columbia Aging Center, my faculty and colleagues there, and thank all of you. Be happy to take any questions you may have.
Okay, Addison, thank you so much for your talk. I was taking too many photos for your slides, but anyway. So one question it's more like a discussion. So do you think, is there any possibility we can reverse the immune aging system, like the process? Because I feel like for this kind of biological aging clocks, we don't really have a lot of evidence that it is reversible, right? But I'm not sure whether when we think about in general this immune aging part, is this really some discussions people are thinking about?
Yeah, I mean, there, that there was at the beginning, there sort of this paper that was really kind of like a commentary that was published in Nature, where they were talking about this, like can we reconstitute the thymus, for example? Can we take these naive cells and transplant them so that individuals have, more naive cells? So I think there's roots, but it's so complicated. Like just, working on the immune system, it's like a whole concert of things that are happening. So I think, some of these efforts, like for example, there was a whole thing when C-reactive protein was discovered as this important inflammatory marker, which, by the way, is an immune marker and was related to infection, which kind of got lost in the whole discussion of CRP as it was integrated into research. We've seen that there's been some really untoward effects when we try to target inflammation, because inflammation is important for the immune system. Once we tamp it down, you have these other, aberrant effects that influence your health and immunity. So I think it's going to be complicated, but maybe slightly more targetable than biological aging. I'd love more Lauren's thought about that too. Yeah.
Okay. Thank you.
Thanks for that great talk, Allison. So as someone who has a way better understanding of the immune system than I do, and also has expertise in cognitive health, a question that, as someone who uses a lot of DNA methylation that's been profiled in blood, which, as you said, is basically capturing the immune system because red blood cells don't have a nucleus. So it's just capturing white blood cell DNA methylation. What do you think about using the immune system to predict cognitive health? So DNA methylation measures in blood to predict cognitive health or, decline, dementia, obviously way better than trying to get brain tissue, which is impossible. But why could we see something that's happening in the brain in the immune system? And do you think that that's a valid connection? Do you think that we can actually get good predictors of cognition and cognitive health from blood?
I mean, I do think it's complicated. Like that's sort of the whole premise of like a neuron is different than the mixture of blood that you're getting when you take a blood sample from a peripheral blood sample from an individual. I think it's tough. I do think though, the research in this area it was originally thought that the brain was like an immune privileged site altogether. So like when I started and I was working on Alzheimer's disease research, the idea was that the brain is this immune privileged site. There's not a ton of crosstalk between the blood-brain barrier and the rest of the periphery. Now we know that is not the case, right? So we know that that's, we see that emerging, and, the integrity of the blood-brain barrier also as we age changes too. So, whether we're going to get exactly what's, I don't think we're going to ever get sort of the specificity about, in terms of what's happening in the brain, but with the sensitivity of measurement, we may get a little bit closer to understanding at least some of the molecules that we know can pass through the blood-brain barrier to affect the brain. I think there is some potential hope there. But yeah, I think it's complicated. This is a tissue-specific issue. I also work in the area of mental health too, PTSD and epigenetics, and this was often an issue. And so, some ways that people have thought about kind of getting around that is to doing, basic research using neuronal cells, the growing neuronal cells from cell samples that you get from individuals, from peripheral cell samples that you get from individuals, to try to sort of see whether the epigenetic effects or expression effects are similar across those cell types. Yeah.
I have a few kind of technical-ish questions. So you had the slides near the end where you were talking about flow versus salus. Is that I don't know any of these words. Mm-hmm. And that the concordance was not super high. Mm-hmm. Do those come from the same physical samples, or are they different draws? And then so it's from the same samples that they estimated those two things. Yeah. Great. And then and how?
Well, actually, well, I guess it depends on what you mean by the same sample. So there's samples that are taken at the same time.
Yeah.
Right, but they're different tubes, right? Like you have your tube, your purple top tube for your genetics, and then another tube that you're going to use for your peripheral blood mononuclear cells.
Okay. And then what's the the retest reliability? If I measure flow twice. Mm-hmm. But from tests taken at the same time, how concordant should I expect that to be? Is it pretty high, or is there some variability?
Yeah, we didn't have. I wish we had an IIV in our study. We didn't have an IIV in our study, where you're basically kind of doing the same measure on an individual at multiple time points. But just from sort of looking at the literature, it does seem to be pretty consistent because it's such a kind of it's a marker that, it's looking at sort of the composition, and these things don't change drastically they change over longer periods of time, I guess is how I should put it. You don't see sort of day-to-day changes unless you're infected, you have an infection or something like that, which we would account for.
I guess I was mostly thinking about measurement, is the not necessarily if maybe the actual truth is constant over time, but even the measurements are reliable enough that we don't think there's any sort of just measurement error.
Yeah. No, I think there is measurement error in flow. I think that's the case with all. I don't know any procedure that doesn't have any measurement error. I think there's measurement error there. I guess the question, your question is your measurement error larger?
Because you said 0.5 for the the killer,
the natural killer cells. Yeah.
So what would I expect it to be if I just did flow twice? Is that going to be close to 0.5, or is that going to be close to 0.9? Or
I think it would be probably 0.5 or higher in flow, I would guess.
So flow versus flow.
Yeah.
Okay, so 0.5 is actually really good in that case. Okay.
Yeah.
Great.
So apparently, a lot of people are interested in using DNA methylation to quantify biological aging. But I understand the point that you argue that some of the patterns we see in DNA methylation data are not nuisances. They reflect, for example, immune aging. But I wonder if the goal is to find the best omic predictor for immune aging. Can you comment on the use of DNA methylation versus some of the other data people can collect, such as just gene expression?
Yeah. Yeah, I mean, depends, I guess, what your question is, right? In population research, I think the gene expression part of it gets a little bit more complicated and maybe more difficult to map out. But I know that, there's other things that you can do too with immune. So these are I would guess maybe I can explain it this way. I would think of these as sort of static measures. They're not truly static, but the way that we are measuring them is static. But there are aspects of the immune system, for example, you can prime cells in a culture and identify sort of the expression of cytokines that they may have. So there's actual functional assays that you can do too. But it is more complicated. It's just a more complicated route. So this is sort of just telling you like a snapshot. Okay, this is the sort of surface markers, the composition that a person has at a given time, but it doesn't necessarily, we are missing some information. It doesn't tell you sort of how competent is their immune system, right? In addressing like if they were exposed to an antigen or a mitogen, for example, like how much does that T cell release some cytokines or, inflammatory mediators that are important for your immune response? We don't know that.
Let me just ask a very quick follow-up then. So is there a specific reason to prefer DNA methylation for studying immune aging?
I don't think there's a specific reason for it. I think it just gives you, as I mentioned, a nice snapshot when we're talking about population level data of what's happening. We know from flow data that these kinds of measures are important in terms of mapping immune aging changes over time. So the methylation allows you to get to those same measures that you wouldn't have been able to get to otherwise in population research. But that doesn't mean that there's not other ways of measuring the immune system and thinking about it. Like I'm not at all against trying other approaches. It's just that epigenetics is being tested in many, many studies, and it now opens the opportunity to understand at least some aspects of the way that our immune system ages.
I've got two questions. The first is pretty easy. Why are you so focused on T cells in this and not looking at like B cells and markers associated with those for aging?
Yeah, no, B cells are really important and interesting. We haven't seen really robust effects of the social variables on the B cells. And I'm not sure exactly why that's the case, but it does appear that the T cells are more sensitive to the social environment than the B cells.
Cool. The other question that I had was about the social implications on the immune aging. And I was wondering if you have a way to kind of differentiate mechanistically if you're looking at, well, there's an increase in these positive relationships with people and there's like a mental health factor or, a CNS factor mediating that versus you have more social interactions and then you're exposed to more antigens and your immune system is responding to that.
Yeah, it is really hard to parse those things out, but I think it's like really fascinating, right? Too, because, we also do research on infectious diseases and we often look at these kind of chronic latent infections and like cytomegalovirus, for example, which your T cells actually become very clonal to CMV. Most of us are exposed to CMV, usually in daycare settings and young age, and then we carry the virus for our entire life. And it remains in a latent state, and it continues to kind of prime the immune system over time. And usually in people who are immunocompetent, you won't see a sort of clinical, even if you reactivate your CMV. For example, there's been, studies that have been done among students who are taking their final exams, and they show that, before they take a blood sample before, during and after the exam, and like during the exams, their CMV levels get really high. There's no clinical aspect characteristic of that at all. It's just subclinically, you can see their antibodies rising. So I think, it's an interesting part of it because in some ways, if you're more social, you're probably more likely to get exposed to more infections, but that's also probably socioeconomically stratified also. And so, I think it's hard to kind of parse apart what the role is of the infectious diseases, but you may be more likely to be exposed, but maybe less likely to reactivate if you have better social relationships over time. So that may be protective for your immune system. But I think it's interesting, just hard to parse apart. I have a colleague right now who's working on including something called VIR-Scan, which is a methodology that scans for all sorts of pathogens in the blood. So you can kind of look at, all the antibodies to multiple different infections that a person may have been exposed to. And it might help you answer some of those questions, like what's the background of your immune system? Because you're exposed to all these infections all the time. How is it modeling biological aging? How is it being changed by the social environment? So I think some of those methodologies will help us answer these questions, but they're open questions right now.
Yeah, I have a very general questions. Like you are focus on the epigenetics of the immune cells. Is there any specific reason that you want to focus on the immune of the aging part? Since like there are other parts like reproductive activities or other cancers, agings that do you think like it's because of the immune is more relevant to the sociology or economics? You think?
Yeah. So I think of it like this. The immune system is very sensitive to the social environment. I think that's one part of it in terms of my interest. But I also sort of see it as this kind of barometer of lots of other things. So our immune system is important as it relates to cancer, for example. It's important as it relates to cardiovascular disease. It's important as it relates to diabetes. Like there's many ways in which the sort of like immune system kind of acts as a barometer for many other outcomes too. So it's in some ways a nice general marker of other factors that might be impacted as it relates to your health.
Okay, thank you.
Yep.
May I ask a second question? But just be very quickly. Maybe this is just an open discussion. But when you think about it in social factors, between these physical factors and the mental factors, which one do you feel like is more important? So the example I'm thinking about is like you can do a lot of like physical activities, right? Like slow running, jogging. But on the other hand, you can also keep like a very open, like a good mood in general, right? So there will be like this kind of in general two different possibilities to positively influence your immune system. Is there any kind of research to try to quantify like whether this kind of physical part is more important or the mental part is more important?
Yeah, that's an interesting question. We've talked a lot about that. Like, is there a way that we can sort of find like which factors matter most? And again, I think it's a hard thing to parse apart. I would say that, physical activity is really important for the immune system, but a lot of these things, again, they're socially patterned. So when we start thinking about some of these behaviors, like you think about air pollution too, air pollution can impact your immunity. But again, that could be socially patterned, right? Where you live impacts whether you're exposed to air pollution, for example. So I think it maybe helps to kind of think about it like, you have this social measure. What are the, it's a contextual measure. What are the maybe more, downstream factors that are related to these social variables? And I think physical activity is probably one of them. And so when you adjust too for those things in your analysis, you might be adjusting again for a mediator that impacts immunity. And just for fun, physical activity is good for your immune system for sure, but too much physical activity can actually suppress the immune system. So you have to, and that's like, I'm talking about like training for a marathon kind of physical activity.